pointer somewhere? Oh, I see. So I give it to... Ja, einfaches Modell. Okay, aber die Leute stehen ja hier. Ja, ja.
Good morning. Good morning, dear Nestlé Nutritional Council, distinguished speakers, guests, and colleagues. Thank you for joining today and participating at the 11th International Nestlé Nutrition Symposium with the topic of nutrition and the human gut microbiome. We had this time almost surprising number of requests and are reaching a size limit with the symposium where we had to decline a few of the requests already. And on the one side, I hope it reflects the quality of those symposia, but it definitely also reflects the topic itself. It is a hot topic, a very wide publicly discussed topic. And with all the advances in science in a multitude of different disciplines and approaches, there is also a lot of lay discussion out there, and some skeptics are coming up already talking about the possibility or the risk of the field drowning in its own success or in its own hype. And I think today we will see with the 2 days we have, they're taking a right approach to the field, looking at it from different angles, which we will do today in 4 sessions, today and tomorrow, going from the facts and figures from the basics around microbiome, right into also clinical, physiological aspects, looking at it from a system biology perspective. There is a good way to early on find good applications of this very interesting and exciting field. And I'm very glad that we again have a really outstanding set of speakers that will share with us their insights, their thoughts, and discuss with us. And I think we will have with each of the sessions a possibility to have questions, to have discussions. I know a lot of you in the room have a background or actively contributing to the field, which actually also Nestlé does. More than 30 years we've been working on individual microbacteria, probiotics, more than a decade already, ongoing research in the field of microbiome. And so we have, as a company, also a very vivid interest in the progress this field makes and try to contribute as much as we can to it. We are, of course, not only interested in microbiome and invest in this field, we're also very interested in your safety while you're in with us, your safety and your health. And before I hand over to Josef Penninger, who is the director of the IMB in Vienna and part of the Nestlé Nutrition Council, he will give an introduction to the field and host and chair the first session, as the other sessions will be chaired by Nestlé Nutrition Council members. I want to have your attention just shortly for a video, a few asks that I have as your hosts to ensure or contribute to your safety and health while you're here. With that, I wish you a very interesting, insightful, and inspiring 2 days. There will be enough time for discussion and exchange. I hope, like the many years before, many of you will with having new inspiration for their own research and contributing even more to the field. With that, let me open this symposium, a short safety video, and afterwards I hand over to Josef Penninger. Thank you.
Hello and welcome. Before you start your visit, we'd like to take a few minutes to make sure everything goes smoothly. Use common sense for your own safety and that of others. Be mindful of what is happening around you at all times. You need a badge to enter the NRC. Please make sure it is visible throughout your stay. To access certain zones, especially the laboratories, you must wear specific protective clothing. Thank you for following your host's advice. We have strict rules on confidentiality and privacy. Don't take any photos without prior permission. In the interest of everyone's safety and well-being, smoking is prohibited at NRC except in designated smoking areas. Constant vigilance is essential. Don't run. Don't use your phone while walking. And please hold the handrail when using the stairs. Misjudging a situation or overestimating your own abilities can have serious consequences. If it looks unsafe or you are unsure, stop immediately and contact competent personnel. Despite all these precautions, accidents may still happen. Remain calm and immediately raise the alarm by dialing 999. Internal safety teams will arrive as soon as possible. Don't panic if we need to evacuate the building. Listen to the audio messages broadcast over the public address system and follow the instructions. Take the emergency exit and head to the assembly point immediately. Everyone benefits from a safe workplace. Inform your contact person of any risks you have identified. Help us to take care of the environment. Don't waste natural resources. Recycle as much as you can. We rely on you to respect these safety and pro-environment-based behaviors. Thank you for your attention. Enjoy your visit.
So, good morning, everybody, and welcome to this meeting on the microbiome and the intestine. I cannot avoid showing mice at the beginning, and so a big thank you to the people who organized this, Thomas Beck, Johannes Lecoutre, and of course the team, and of course to Nestlé to support this meeting. So it's very important that a company like Nestlé is being exposed to the latest science, have a critical discussion about some issues which have been developing over the last years, and of course nutrition and microbiomes and how microbiomes and food influence health and immunity in general, of course, very important. So I wanted to start this a little bit with a personal anecdote. When I started my own laboratory many years ago in Toronto, before I moved back to Europe, I was actually on a PhD committee of a young student, and he was studying diabetes and obesity in a particular mouse strain. So they had published lots of nice papers and had described nice phenotypes, and all of a sudden the phenotype completely disappeared. So for 2 years, nobody in this laboratory could actually reproduce any of the findings which had been done before. So this was, of course, a very serious issue for me, being a young faculty member of the University of Toronto. So what was going on? So this was in the mid-90s. And the solution to the problem, actually, was 2 years earlier they had changed the water supply. And the supply of water was actually changing the pH of the water. And the pH of the water, we would probably now all conclude, changes the composition of the microbiome. And this was actually the solution of the issue. So they changed the water back to the old one, and voila, next day the mice got diabetes again. So this was kind of an early entry into something which we at that time believed we can solve everything with genetics, and we just make one knockout mouse after the other and we can explain the world. And of course it was a little more complicated. And of course, for me also as a trained immunologist, we always knew that, for instance, NOD mice which are susceptible to diabetes, have very different phenotypes in different laboratories. In one laboratory, they got it very early, and the next one, they basically didn't get it at all. And so I think over the years, we came to grips with that there's something more than genetics. There's something more than epigenetics. Is another important organ in our body, which is, of course, sculpted by nutrition, water supplies, and many, many other little things. So just to give you a little introduction to the intestine, and this meeting, I think, will be spectacular. All the speakers we lined up and have been lined up by Johannes and his team will give you a very good insight to what's going on in terms of defining microbiota, what they might be doing in health and disease, how this is changing over time. Just to give you a little idea, so, you know, with 3 of us, we could probably play a nice soccer game. The guts of us, because it has quite some large surface. And I just want to show you this movie because I love it, from Hans Kleber's lab. So over the last years, we learned a lot how the gut is being built. One can actually clone and find the stem cells. And by work from many people, and from Hans, for instance, so in the small intestine, the villi, and then one can fly into the crypts where there are stem cells. And of course, every day in our intestine, there are many cells which die and many cells which are born in this little place here, where the stem cell, LGR5 positive cell, and they somehow clonally expand the intestine and basically have a conveyor belt of new cells. One can even take them and clone them and make little guts, so-called mini-guts, mini-organoids, which of course are really interesting for research. One can make a pancreas now out of them, a liver out of them, many other tissues, and at the end you end up with this beautiful little thing. And as you might see, there's even Nemo swimming down the road. I think I need to show Nemo again. So that's really Hans Kleber's work, you see, the little guy. So that's basically what we're dealing with, a very complex tissue with and of course, these cells don't live by themselves, so they live with the topic of this meeting. There's probably 1 trillion, and we might hear about the latest data from our speakers, Willem de Vos and Dusko Ehrlich, about latest data. So there are about probably 10 times more bacteria living with our guts than in our total cell numbers, and they, of course, influence host metabolism. There's some data that influence behavior. There's some really cool data in Drosophila, another species I love. Apparently, the bacteria in the gut influence sexual behavior, so mating behavior, preference of mates. So be careful what you eat in the evening here. Of course, health and susceptibility to disease. And over the years, of course, people have figured out there's this microbiome living with us in the intestine contributes to lots of genes. And as Gary Rafkin once said, these bacteria living with us are true chemists. They can do spectacular chemistry which we cannot do, provide us, of course, in mutualism with lots of advantages, breaking down food components, helping to provide vitamins for our body. And of course, this microbiome, and Susanne Suter will probably give you a much better introduction to this, is being sculpted by life experiences. Of course, the first big life experience is birth. So there are probably differences between vaginal birth and birth by C-section, because part of the early microbiome is actually coming from the vagina of the mother when the baby is born. So these are very important issues of health, probably in the future, by breastfeeding, particular sugars which are in the milk, which sculpt the microbiome. Of course, the food we eat, sculpting the microbiome. And this is, I think, a very important issue for companies like Nestlé for processed food, because, of course, over evolution, we as a population have basically not eaten processed food. We have eaten food which was spiked with bacteria. And for instance, in this example here, in seaweed, there are certain bacteria which people have always been eating if they're not being processed or not inactivated. And this bacteria, actually, it can transfer the DNA to bacteria living in our intestine. And by doing so, there's this permanent gene transfer going on. And of course, the microbiome then is endowed with new DNA, endowed with new chemical capabilities. In this case, endowed with the capability to break down particular sugars from seaweed. So basically, during evolution, we have been permanently eating bacteria, which then have been bringing in DNA into our microbiome, and by doing so have sculpted us as we are, and of course has sculpted our health. And of course, an important issue to consider in terms of processed food. And of course, the microbiome is sculpted over time, and in the babies by breastfeeding, formula feeding, in the little toddlers, treatments of antibiotics, nutritional preferences in the adult, and of course being sculpted in the elderly. So there are, of course, very interesting work going on, how these changes occur, what influences these changes, which part of the microbiome is stable, which one can be changed. And we will hear lots of excellent talks about these topics. This is just a very superficial introduction, so forgive me if I'm missing something. And there's, of course, another issue, and this is, it's not just the bacteria which lives outside and then in the gut lumen and then, of course, intact with the epithelial layer, but they're, of course, very important to sculpting our immunity, and, of course, immunity meaning our reaction to allergies and food tolerances. And what I find really interesting is that 70% of our immune system actually lives in the intestine. So they don't mostly live in our spleen or blood or lymph nodes, but the intestine is actually by far the largest immune system. So the P cell, the T cells, the plasma cells, the antibodies, the regulatory T cells, the very unique innate immune cells. We will hear, of course, later in the talk here. And of course, there's this permanent interplay between the gut microbiome, talking to the epithelium, talking to the immune cells, and by doing so, changing immunity and sculpting a healthy immune system. So this is how it basically looks. It's a beautiful image of so here's the epithelium and the descriptor patches, the dendritic cells, the various subtypes of T cells. So a little bit about something I have been doing over the years. So many many years ago, we actually cloned this gene called angiotensin converting enzyme number 2, doing flight genetics. So no worry, we are not there yet in genetic engineering to make flies with teeth. So we are getting there, but it's still a little complex now. And over the years, we figured out, it turns out, to be a master regulator of the renin-angiotensin system, which controls our blood pressure. So if you get up at break, this is the system which modulates your blood pressure, heart function, kidney disease, which was quite cool. And then we also figured out that it's actually the essential receptor for the SARS coronavirus. So this is how the SARS virus actually enters our cells, which is also interesting in figuring out some viral entry receptors. So this is actually the SARS virus. Our knockout mice, the virus cannot replicate. And then we realized and found a paper that the SARS virus replicates in the gut epithelium, which didn't make any sense. And so we started to actually look if this protein we had been working on for 10 years, controlling heart function and blood pressure, might be actually also expressed and be present in the gut epithelium. And amazingly enough, it's actually at the outside, at the luminal surface of the gut epithelium. So it's highly expressed, this little protein called ACE2. So this begs the question, what is the master regulator of blood pressure control actually doing in the gut epithelium? So what it's doing there. And turns out ACE2 controls expression of particular amino acid transporters. So it had a completely different function as described in control of blood pressure. And actually what it does is it controls single amino acid transporters, and by doing so controls the uptake of particular amino acids from the diet we eat. And one of the amino acids is actually tryptophan, an essential amino acid. And as a consequence, these mutant animals actually have reduced levels of tryptophan. And as a consequence of this, what we actually found is these mice are super susceptible to get inflammatory bowel disease. And so the question was then, could this actually explain, or be one of the explanations, how malnutrition in people who don't get enough to eat, people who have anorexia, could this actually explain why this leads to inflammation of the intestine and diarrheas? And so what we actually also did is we developed a diet for animals which was tryptophan-free. And when you do this and trigger the colitis or intestinal inflammation with this little drug here, then the animals develop super severe colitis. And these mice develop very severe colitis, and when we actually substitute tryptophan again with a D-peptide, because this D-peptide transporter is still active, then we could basically wipe out colitis again. So, and the reason was, turns out that this molecule and tryptophan regulate the ecology of the gut microbiome. And I will put it all together, no worry. So we made it actually on the cover of Nature, because in essence what we had found is that this is a very important molecule, how dietary amino acids are taken up in the intestine. And this then couples to the immune system in the gut, to innate immunity. And because innate immunity is changed because of the change in diet, because of dietary tryptophan, the bacteria change in the gut, and because the bacteria change in the intestine, these animals become susceptible to inflammation. So we believe this could be one of the explanations. There are probably many explanations out there, how malnutrition leads to inflammation of the intestine and diarrheas, because if there's not enough of the essential amino acid tryptophan, innate immunity changes in the gut, the microbiome changes in the gut, and of course all this data on Kwashiorkor changing the microbiome changes, and this of course leads then to susceptibility to inflammation. So this was from a recent review about our work. So this is ACE2 controlling this amino acid transporter. Of course, tryptophan-rich foods. I'm sure tryptophan is just the surface of many other food factors which could do very similar things. Influences the gut epithelium, the microbiome here, and it also influences via metabolites which are being generated downstream of tryptophan, they directly influence the immune system. And probably at the end of the day, where this work will all go is that we might figure out over the next years and decades how particular food groups change and are being metabolized in the intestine, how these metabolites then actually bind to particular receptors on particular innate immune cells or gut cells, how they feed into the microbiome, and of course by doing so, these particular nutrients then would sculpt immunity, and by sculpting immunity and, of course, metabolism and many other functions, sculpt health. So I do believe slowly we can move into the direction of a molecular description, how food and nutrients couple to immunity and couple to metabolism and health in the entire body. So this is, of course, only the surface of it. So recently there were nice papers published in Nature that another product from the gut microbiome called butyrate can directly change, for instance, regulatory T cells. Dan Littman has beautiful work published that particular bacteria in the intestine control a cell type called the T helper 17 cell, which might be a link between gut microbiomes and arthritis. So if this is real, then one could actually, with nutritional interventions, reduce incidence of arthritis, which would be spectacular if possible. But of course, there's lots of work to be done. Just to bring this back to nutrition and of course put in a larger context, is this recent studies just published that artificial sweeteners change glucose tolerance. One can, of course, see the data as one wants it. And it's, of course, a very interesting issue. And just another anecdote in the school of my 12-year-old daughter, for instance, they tell them, please don't drink Diet Cokes and soft drinks with artificial sweeteners. So this is kind of already filtering into the schools. Last week I was invited at the microbiome meeting in Toronto at the Hospital for Sick Kids. In all the breaks, you cannot find any diet soft drink anymore, so they just serve soft drinks with all the sugars. So I think one has to face these issues because they're published in Nature, they're discussed in the popular press. And from a former student of mine, he actually did a very similar experiment. It's not published yet in Drosophila, in fly, and it changes their behavior. So I think these are issues we have to figure out. And of course, it needs a lot of research, a lot of work, because at the end of the day, there are many, many questions. Not just this one, but there are questions of can we ever figure out the microbiome? Do the work in groups, or can we find a singular bacteria, for instance, which we could modulate, put in a yogurt, and then we can modulate incidence of arthritis? Can we feed newborns particular bacteria to give them anti-allergic effects for the future? I think there are lots of issues. The issues of what's the chicken or the egg? How can we really establish in the complex tissue like this with thousands of different bacteria, how can we establish causality? How do we really know that the particular subgroup of bacteria is responsible for obesity, for instance, or is actually obesity changing food uptake and nutrient uptake, and this is the reason why the microbiome then changes. So I think there are lots of questions, and some of the questions will be discussed here, so I think it's a very, very exciting time. It's a very important issue for a company, for Nestlé, and of course for global health, with grand opportunities, because if one figures out how nutrition, particular food groups, feed into an intestine, give us healthy intestines, give us change our immune systems and against arthritis, of course susceptibilities against cancers, behaviors, then I think there's a very intriguing way to possibly make a major impact on the health in this world. So with this, I stop my little introduction and introduce our first speaker, Willem de Vos, who studied biochemistry and obtained a PhD at Wageningen University and also at the Max Planck Institute for Molecular Genetics in Berlin. So since 25 years, he's a professor at Wageningen University, also holds a chair in microbiology, and is a professor at the university, works with the Academy of Sciences in the University of Helsinki, has won many, many prizes. The prize I I like is the Netherlands Most Entrepreneurial Scientist Award. So maybe you can tell us about how you can mine the microbiome for new business opportunities. I think there are many, also great chemistry one could work on. And of course, he has made major contributions on the molecular composition of the microbiome, on the metagenomics of the microbiomes, on the whole system of it. And he, of course, has made major contributions on microbiome in the intestinal tract of humans. So it's not just mice, my favorite species, or Drosophila, but his work has actually a real impact on human health. And so thank you so much for coming and telling us about the work here.
Thanks very much. Okay, thank you very much. It's a pleasure to be here and a privilege to stand here and talk to you. I thank all the organizing committee and Thomas and Stefan and Ed to get me here now and then and talk about something which is close to your heart. And for the business people, close to your business card. About 10 centimeters from your heart is the largest community of microbes. And I'm happy that Nestlé allowed me to put my logo on the title, which is Microbes Inside, and it introduces the subject. And I have to say to the chairman that don't believe everything what is written in Nature. So it's good to come back to that later. One other thing is that we live in a microbial world. You know, you and me, we're not alone. We have about 10 to the 30 of these little creatures on this planet. That's more than there are stars to be calculated in the universe. So it's good to keep that in mind and be a little bit modest now and then. When I talk to the students, I say, well, 1 spoon of GI-tech material has more cells than your brain. So it's good to keep that in mind sometimes. And they have an impact on all kinds of areas, and you can see here all the biotech areas, and maybe that's the reason to explain this activity I had in the Netherlands to set up some companies. Because white biotech, you know, marine biotech, agro-food, animal, pharma, they all use microbes. But we use them too. They're very important for us. Actually, you can put it on yourself, and that's the impact of it actually is illustrated in this little cover of The Economist. This was a couple of years ago, but still, I think it's important to illustrate this, because the title is Microbes Make Us Man, but actually, you know, it's Microbes Make Us Man and Manners. It's good to remember that, because we know now that our gut also communicates with our brain, and I'm sure that other people will come back to that. If you read around this cycle, you can see that how 90% in the cells of your body are bacteria, the benefits of fecal transplantation is other things you would rather not know before breakfast. So therefore, I'm happy that I speak now to you about this, and I introduce the subject without further, discussions. I also can say that we have found and published recently that in some cases, not in all, in some cases, bugs are better than drugs. And I will come back to that later in the C. diff case. So what about the economy? I know there are some economists here, people who like to know how you get money out of this. And I'm very happy that they are here, because if there's no money gained in this area, we can't do the science. So it's good to have that going on. So there are attractive market opportunities, as a recent market review actually indicated. I was a bit disappointed, market value of 250 million, increasing to 650 in 5 years. That looks good, of course, because 22% annual growth. Well, I think many of you in the audience would like to see that. However, I think when you think about it, it maybe is underestimated, because I think this area will have a bigger impact on consumer and patients, which you can't even express into money. The daily, the DALYs or the health, I think you can calculate into dollars. It's good to keep that in mind. I think this area may grow even faster. And maybe after my talk, or maybe the end of the meeting, you will have the same idea. Let's come back to what's happening there. It's more than only food conversion that microbes are doing. The conventional belief is that, you know, our diet, we take it up, we take the goodies out of it, and the bad, the difficult things, the non-digestible food components are converted by the microbes in the colon. And they produce a little bit of short-chain fatty acids and a few other anthracides. And that's sort of it. It's more than that. It's much more than that. Why is that? Because they talk to us, these microbes. They have specific interactions with us. And we will hear more and more about that in those days. And they also have evolved with us. They have this mutualistic interaction. They're not competitors anymore. They are symbionts. Actually, in the old days, they were called commensals because they eat the same thing as we do. But now we actually have this mutualistic interaction, so they're more than that. So it's good to keep that in mind. There's a little friends inside. Keep that on your retina. So why is this human gut microbiome so hot? Nowadays, there are more than 1,000 papers per year. Actually, every day there's something to read, and also in high-impact journals. One of the reasons is that sequence has become mainstream. We all can sequence the world and look at that. But we also have to realize that we are in the beginning of the discovery tour. We have such a limited knowledge and limited concepts, and it's a very complex system. I will show you why that is the case. So it's not so easy to make progress here, especially not in human. But it has an unprecedented health impact. I mean, it's very important for our health. This is a little cartoon I made with my daughter, Lisette, and she's a medical doctor. So she helped me by looking at all the diseases because I'm a simple biochemist. So it's good to keep that in mind. And we calculated there's about 20, 25 or so associations being presented to be associated with microbiome. Therefore, the title is From Correlation to Causation, but still correlations can be important. We come back to that also later, I think, in these presentations, because it's not only consequence, it can be correlating and can also be very important. But it's good to think about the causation too. It's not only, a few real examples of causation. C. diff I will discuss today. Metabolic syndrome, I'm sure that Max Nierder will come back to that tomorrow. What is the problem? The problem is that most of the microbes have not been cultured yet. And not only in our gut, but everywhere in the world. So there's all kinds of programs, not to look at the microbes themselves, but look at their proxies, like DNA. DNA you can sequence, I already alluded to that. Next generation sequences, third generation, whatever, single molecule sequencing, we all can do that now. Actually, I was interested that Josef described my CV. I did my first sequencing experiments when I was an undergrad student. And I sequenced the mitochondrial DNA. Later it turned out to be bacteria, so I was on the right track already. And that day, we could publish that. One little sequence. That was 30 years ago. I published my first paper, or 35 years ago even, I think. So it's a long time ago. So I've been sequencing. I know what DNA is, and I love all the things we can do with it. But we have to be very careful also not to overinterpret, because I'll show you also why. Now we have this 1,000 of species in the gut. You can calculate how much there is. It's also a matter of definition. Some of them are very hard to culture. So we have to be also modest there. But we can use this molecular microscope and use the DNA as a readout, and also the proteome if you want, and other high-throughput technologies. And then we come to the idea that there's more than 10 million genes in the gut, and other people will come back to that. I'm very happy to acknowledge the MetaHit proposal and the project, which came also to the cover of Nature, and Dusko will elaborate on that later on. We can culture. We can culture quite a few species. Actually, we made an inventory recently for the first 1,000 cultured intestinal microbes. You can see there are fungi. That is our eukarya. There's bacteria and archaea there, but the majority are bacteria. So we will say microbes, they're bacteria, basically. And if you look at the development, you can see that this was the first notion that there were bacteria in the gut. Actually, that was Escherichia coli discovered. It took a long time before really a new, I would say, leap in the activities came. That was when the anaerobic technologies were developed. Hungate papers, and actually, you can see that immediately materializing. More people were doing this. Why is that? Because all the microbes in the gut, or most of them, are strict anaerobes. And now with the high-throughput systems, we can do more. And we can extrapolate that, and I know of people doing that in a real fantastic way. And I think we can crack that problem. So we continue to isolate novel genera, we pull them out. Actually, I'm interested in not having the inventory of all, but I look at the major players. So we look at Akkermansia as an abundant and specialized mucus jugator. We'll come back to that later. We pulled out Ramboutia as an ileum-adapted butyrate producer. Was already introduced why butyrate is important. Another one is Intestinomonas, which is a canonic one. So we pull out new organisms all the time, and we try to understand what they're doing. And I like that, abundance and function, because that's the main drivers of why they're important. Now, what is the problem of this whole area? The problem is that you and me, we are different. We're different in our genes, we're different in our microbes. Actually, we're different maybe more in our microbes. We found that out already about 15 years ago when I started this field. Actually, I was more in the white biotech and the food biotech, and I started to work on the intestinal tract because I saw there are many microbes there. We don't have to go to the moon or to the deep ocean to find microbes. We can find them just, very nearby. So we looked at that with Arizona, my first PhD student. I found in a dozen subjects that they all had a different microbiome. So you and me were different. Well, we expanded that. Now, there's 3,000 people here. We have 10,000 in the database. This is a picture of 3,000. There's a complex PCA showing that each and one of us has a different microbiome. However, when you look at monozygotic twins or family members, you can see they're more similar. I will expand on that a little bit. This is work done with my co-workers in Finland. You can see the Finnish names here. If you see them, that are the bioinformaticians, biostatisticians who work with me to crack this tough nut. This is the twin studies we did. A long time ago, we did it already. People didn't believe it so much. We repeated it as well. 40 monozygotic twins, and you can see monozygotic twins. The pairs look more similar to each other than to the unrelated individuals. Not all bacteria are similar. As you can see here, some actually are more similar in the twins than others. I won't go into detail, but it suggests there's some specific interactions. And maybe there are specific receptors that make those organisms stay. It's not only colonization, because those are adults living in different households. So this bacteria has stayed there for the 30, 40 years of their lives. We find this quite often. Not in all countries of the world you find that, because in those cohorts in Belgium and Holland, there's limited antibiotic use. The people have the similar diet and they don't move so much. So in a way, that's also maybe a factor which can be confounding and also explaining why other people can't reproduce that so much. But it tells you, and it will give you other data, that we have this intimate interaction with our microbes. And it's related to our families. Now, what about diet? If you look at the dietary compounds, they have an impact on their microbiome. However, this personalized microbiome is more prevalent, more important. So if you put a percentage on it, it's more personalization, subject-specific variation is more important than the diet. However, the diet is important. This is a very low-cal diet. People are losing weight like crazy, and you get a large impact on the microbiome. This is a fiber diet. I will come back to that. It's only a few percent. However, it can be very important, few percent. Don't misunderstand me. I mean, it can be a very important fraction of the microbes that are changing, and they may have an effect. I will show you that later too. Probiotics don't do much, but as we have seen also, they do more in the upper GI tract, immune regulation, and we saw already lactobacillus on the slide of Josef. So the personalization actually overrides the fluctuation of a normal diet, which is good, because you and me, we're different. And we go and have our lunch here, or we go and have McDonald's food for a couple of days. We're not going to have the same microbiota, which is good, because we have to have this flexibility. And we have to have the personalized links, because the microbes link to our receptors probably. They have this special mutualism, this interaction of symbiosis in your gut and my gut, and maybe a bit different. Of course, the principles can be the same, and therefore we'll look at the mechanisms. Also, your microbiota is stable. We follow people for more than 10 years and found they're very stable. So it's good to keep that in mind. You can actually recognize people. We used it for a court case too. Now, we are different, but we're also the same a bit. There are mechanisms there, of course, that rule this system. Mechanisms that explain the enterotypes that we found in the Metahyte project. They're high-level ecosystem solutions. This is 5,000 people, and Jarko made a nice picture of that, showing that they all separate into clusters, and you find your favorite microbes here of the clusters. It allows you also to stratify subjects. I will come back to that later, too. But there was something else we found. We took from this data set that we had about 1,000 healthy subjects, because we wanted to know what's happening in health. Then we looked at all the microbes we could find, and we found something special. Some microbes have this bimodal distribution. In some people they're high, and others they're low. That's funny. That's something which we started to work on. Actually, it suggests there's some feedback loops, because they're kept in that situation. They don't fluctuate, because many are fluctuating, but others are not. It suggests that there are positive feedback loops, because there's an autocatalytic event or runaway situation. They stay in the same position, they don't flip back. That actually is reminiscent of all the stable states, which have been formulated in ecology. Actually, that was work of Martin Scheffer. He works with us in Wageningen, and we looked at, he actually published this paper as a review about the catastrophic shifts in ecosystems, varying from the Amazon to the ocean to the Sahara vegetation, whatever. You can apply this model everywhere. And what it is that you have a species in a stable state here. This is a little ball here. And its state is resilient. It can't move, as you can see. But if the conditions are changing, so if your diet is changed or you use antibiotics or some other factors, it may change in such a way that the perturbation can get it over this hill very easily, and it flips. The fact that you have this in healthy people, this minimum here suggests, in this bistable, bimodal organism suggests we have this kind of alternative stable states in the gut. You can see them here. You can see here Prevotella, which is quite abundant in healthy people, and Fragilis is the other way, actually it's very low in healthy people, but it goes up in diseased people. Prevotella goes down in diseased people. And this is another one. They're uncultured yet. Uncultured coccidialis, actually, they actually are a little bit varying. But we see if we expand this to cohorts of individuals which are compromised, we can see it associates with obesity and IBS, for instance. So we can use this system now, and this gives a sort of basis paradigm for how we can interpret this gut. It's maybe not so complex as we think with thousands of species. Maybe we have to look at the bistable ones and try to understand what's happening. Now, I heard from people that I speak fast. So I thought, well, they usually ask me for as a closing lecture, because then everybody has to go to the plane. But now I keep you to the coffee, so I have to summarize. So I'll summarize a little bit here. For the people who lost track, they can hook up again. So here we have health and disease. So the conclusion is we are different. We have a personalized microbiome. Our microbiota is affected by diet, but we have limited insight. But new concepts are emerging from the data sets that we have. And one of the concepts I want to share with you is the one I developed with Max. He speaks later, and Erwin, actually. We have this little cycle. It's not rocket science, but it's a way to see this. We are here, homeostatic and resilient microbiome, highly diverse. You have to see these alternative stable states. The little ball can't move, the minimum there. But if there's an infection, you get a little bit of Campylobacter after, or maybe when you go to the airport or so. And you have antibiotics, or you change your diet all the time. You have a very high-fat diet for weeks, and you change the lifestyle. It may be that you go to this different community. This stable state is changing. This little ball is changing. It goes over the hill to another state. And that can lead to this early warning signals that we can see already in the gut of many of the people who are compromised. Often it's associated with organisms which make LPS, gram negatives, which may cause low-grade deformation. The same holds for the flagellins, and we come back to that later in the talks. That may lead to tissue destruction or some effects of the body, and it can eventually lead to strongly reduced diversity, catastrophic shifts, as we call it, also in the ecosystems I described in the ocean and the Amazon. That catastrophic shift leads to chronic disease development. The only thing you can do is to restore the microbiota. I put the arrows here reversible, but we think here we are sort of endogenic minimum. You can't get out, you have to restore it. And that's where fecal transplantation comes in, but maybe also other therapies, which are dietary or pharma-related. I will come back to that later. And you can go back to this state again. Now, this is the model. Well, it's not rocket science, as I said, but it's helpful to understand what's happening there, because these early warning signals can be used. Maybe these early warning signals or the homeostasis resilient data can be used to stratify individuals when you want to do interventions with nutrition or pharma, because it's important to have as homogeneous population as possible, in spite of the fact that we are all different. Now, how is the field moving? Here have the yin and yang again about health and disease. I now make it animals and human. Well, for some reason maybe, because animal studies are beautiful, and we can do a lot with that and learn about a concept. Concepts. But I indicated to you, you and me, we have a personalized microbiome. And what do you think what happens when you put your microbiome in the mice? So it's different. So we have to be aware of that. So I prefer to look at human cohort studies and try to understand also what happens there. This human as a test tube organism too, because we can see that and we can work along those lines. Now, what happens in literature or in the field? There's often this binary microbiota comparison. You take a healthy person and a diseased person, and you look at the microbiome. It's not difficult. You take some shit and you take some DNA out to meet it. Everybody can do that. And you find differences. Well, that's not strange in biology. It would be nice if you found the same thing. Because in biology, it's very difficult to prove something is identical. It's very easy to prove something is different. So be very, very careful there. You always find differences. So what do they mean and how can you use that? So longitudinal analysis is useful too, to follow things in time and associate that to development of the disease or impact of diet. It's important to do that. We are there a little in the beginning. I'm not saying that is not a good way. It's a very good way to start, but we have to follow up. And then we have microbiota-based therapies, and I will give you some account of that, because that's the ultimate proof of the pudding, is the eating, so you have to do something there. Before going there, I show a little bit the architecture of the gut. It was already alluded. by Josef. I love this picture of Hans. He's about 25 kilometers away from us, and we use his organoids. This is the ileum, or the short bowel GI tract, and here, things are happening. As you heard about that. The stomach is a sort of sterilization tank where the food comes in first, goes to the small bowel, and then sits for longer time in the colon, and this is all kind of last fermentation, last pipeline solutions, because that's what happens there. About 5% of the caloric load is still taken up here in the colon in the form of short-chain fatty acid that fuel enterocytes. But there are different systems there. There's more nuances to be made. There's systems which are different. There's a progressive colonization. Look at the numbers. This is about, well, up to 100,000, a million-fold, sometimes lower as compared to the colonic microbiota. Goes a bit progressive in the ileum. So the numbers of bacteria are different, and I will show you also the type of bacteria are different. But that's where all things are happening. It's good to keep that in mind. Colon is important too, and everything what's in there should come out. So you can look at DNA and the proteomes because it gives you a sort of memory of the past. But it's good to keep in mind that the upper GI tract is very important because there's the competition with the host, and that's where things are happening. Now, I talk about that. It was already alluded. Actually, in the textbooks you find it's 300 square meters. It's not a tennis court anymore since this summer. As your colleagues of Fred actually found out, it was 30 square meters. So it's only a service box. Well, still is good, huh? I mean, it's not a tennis court, it's a service box, but it's fine. It's important, the most important part of the tennis court. I'm not sure whether you play tennis, but that's how it is. So it's a very important part. And this is where the things are happening. This is where it all happens in this tennis court. Here you find signaling to the GI tract. Michiel is here. We did this work with human volunteers who were eating probiotic lactobacilli and found effect on the gene expression and the whole immune system, I would say, triggering and tolerance, which is affecting there. So it's an important point. Food digestion, nutrient transport, that takes place in the upper GI tract. Vitamin receptors, other receptors are all there. This is where the things are happening. Metabolic immune system, we talked about it already, so you know that. And the microbes are talking to us. So this is an important area. So how to get there? It's not easy. You can go in with scopes. We can also use ileostoma patients. We did different ways of looking at it. And actually, the story is that in the upper GI tract, there are other bacteria. There's less bacteria, as you can see here from the barcodes. Every barcode is a bacterium. This is the colonic bacteria, a lot of barcodes, a lot of bacteria, especially strict anaerobes. They're not present in the upper GI tract, at least not in the same level. They're just tenfold lower. Streptococci and Veillonella are the dominant organisms. So it differs from the colonic microbiota. There's a unique expression paths where I come back to that. And there's some environmental factors which also affect the microbiota composition, especially in the upper GI tract. Depends on your diet. I mean, Michiel has shown that if you take aspirin, you can even see that coming back in the expression in the upper GI tract microbes. Another thing is there's circadian rhythms. I mean, you have to read the Cell paper of the Israeli group that also has this other paper which was exposed. I had some correspondence with them because circadian rhythms are everywhere. Of course, they're also in the gut. We published that years ago already in 2012 that there are circadian rhythms in the upper GI tract. Microbiota is changing. Your morning microbiota in the gut is different than the evening microbiota. It simply has to do with food and food intake. And when mice run around at night, then I also have a different microbiome in the mice. Well, fine, no rocket science. And we found it also with travelers. We published a couple of years ago a longitudinal study where people actually were analyzed for the fecal microbiome. And there we found that you and me were very stable. But we found sometimes less stability. One was using antibiotics, and 3 others had been time zone traveling. So we reported already about the circadian rhythms there. They may affect the microbiome, but if you don't have any tools to work on it and do something about it, it's difficult. So it's good to keep that in mind. I was interested in the next step, how to then go from that to the next step, how to use that in a practical way. So here we looked in a practical way to the RNA stack analysis, to the transcription of the microbes there. So what came out actually is that there's a competition in the upper GI tract between microbes and the host. Well, no rocket science either, because microbes compete everywhere. They compete also with us. They compete for easy to degrade sugars. So the glucoses and so much is there. You have a heap of starch, I mean, your amylase is there, you get glucose there, and the streptococci are the ones that take it up. They're full of transport system for sugars. They compete with us for the sugars. But they make lactic acid. Lactic acid is not always so good for us. It's fantastic to make plastics, but not so good for us. So you have to compare that, to convert that. So you can convert into propionate. And an organism like Veillonella is doing that. It's a very active organism. Others can take acetate and lactate because they also make acetate. Also the lactobacilli do that, especially under a little bit of oxidant conditions, because it's more favorable energetically. Acetate and lactate can be converted into butyrate. But not all organisms are doing that, only a few. So we isolated those and we'll look at them. This is the RNA-seq data, which just illustrates the fact that looking at the microbial composition is not the old thing. This is 2 individuals. And they're actually dominated by the streptococci are in blue, and the red are Clostridia. So they're a little bit different, as you can see here. Now we look at the pathways. Flora was doing that and found that this pathway is green. Well, this green is not almost visible there, but this green is the veillonella. This conversion of the lactate propionate pathway is exclusively done by veillonella. So if you look at this, microbiome data, beta genome, RNA-seq, you won't see it, but if you look at the pathways and zoom in on it, see, well, this is an important factor. This is the conversion which is relevant, because that signals to our host. And I will show you that later too. So that's how to approach this. So it's not so easy. So they talk to us, they produce something in return. Vitamin production is mainly in the upper GI tract, you have the riboflavin, the vitamin B12 and so on, which are very important factors for us. Trophic chains are like here, the trophic chains are producing molecules that fuel our system, but also signal. Signaling was already indicated, I will come back to that. And new adaptive species are there because those organisms live in a harsh environment. Upper GI tract is not so nice. Bile is there, all kind of stuff there, enzymes. You don't want to be there. So they like to be there and they work with us. So we have been selecting them. So I stop here with the binary comparison. I want to go to a longitudinal analysis. This should show you the power of that. And I like to start with early life. Well, that's where it all starts. Maybe even start before early life. We know that it's -9 months, maybe even before, but still before conception even it may start. You never know. But then still it's important. We're born virtually sterile. We colonized progressively, quickly in early life. And I think this is a programmed microbial transplantation. Not so much for the vagina. I'm sorry. Of course, some lactobacilli may be there. I love lactobacilli, but the ones you find in the vagina are not always found in the human body. It's fecal microbiota. All the bacteria you find in your system, they come from your mother's feces. They have to come from somewhere. But everything is everywhere. As I will show you, they will come there. This programmed microbial transplantation gives evidence for that, and there have been these zillions of mother-baby transfers in our history. Our genes are different because we have this, we get them from our father and mother and our family members. But the same holds for the microbes. You can envisage there has been millions of years of evolution and mutualistic interactions evolving. So our microbes actually have been evolved in harmony with our body. So your microbiota is different from mine. And they're certainly different from the mice. Okay, this explains the family impact. Now, looking at the diversity development, this is a data set of about 5,000 people that Leo analyzed, and actually you can see diversity, which is riches and evenness, they go up in time. Actually, we were surprised to see, we are not completely sure this is really true because we have a little bit less people here as compared to the rest, but it starts from almost 0, it goes up very quickly, but it takes a long time to develop. It's good to keep that in mind, more than we think, at least more than the 3 years which is in the literature, maybe 5, 10 years. Then you get this diversity which is steady in time, and it goes down. I have to say this is not a single individual followed in time because there's no funding, for instance, for 100 years. So that doesn't work. So this is a bunch of people that we collected. Here we go, and I will come back to that later, but not much because other people will talk about it. I think Paul will cover that. So what happens in the first week of life? I mean, we're colonized, I will show you. But most of the babies are lookalikes in early life. I will show you that too. They're very similar. They're not the same as the mother. So how then can it be that when is this microbiota going to look like the mother? So we had an experiment I did with Lotta, a PhD student in Turku, with Erika Zelauri, and Sepasamani. They have these cohorts, and I digged in the freezer. I took them out when I was a visiting professor there, and looked at that. So we have baby samples of the mother and the babies and the mothers for about 3 years of time. If you look at the similarity between the mother and the baby, it's very low in the beginning because the baby has a different microbiome, has different food too. There's mother's milk and whatever. Only after weaning, you can see there's similarity coming. And actually the highest similarity we found was 3 years. Well, that's why the sample stopped, so I think I can only show you this. But the crux is, when we looked at the 3-year-old baby and the mothers after 3 years, and we compare them, we could see that the kid-to-kid similarity was much lower as the kid-to-mother similarity, as you can see here. So it's really different, really much higher because this is similarity index is really telling. And if you look at the XY plots, they're almost on one line in some cases. There's also even, they're of course also more similar to their own mother than to a random mother. So it's good to keep that in mind. So this is really different. So really we can see there are differences here. So we get our microbiota from our mother. I think this is testifying for that. However, it's not really evidence. It's not easy to do this experiment. You can do it in mice, and you can see that. But then here, this is, I would say, support of the maternal transfer. But how does it come then that you can't see it in the beginning? Well, this is actually has to do with the programmed microbiota transplantation. The microbes are there already, but you don't see them because it looks like this. This is how a baby looks in time. Every barcode here is microbial species. You see the meconium sends out. It's a bit of a mixture of everything. It has to do with the fact that this is 100 times lower, actually, in terms of DNA. So actually, it's a very low level, but many things are there. If you look later, you can see the bifidobacteria coming there. They're going to stay. Bacilli are coming, going. Bacteroides is coming. And here you see the proteobacteria coming. And this is a typical breastfed baby of Holland, and you have hundreds of them. Here you can see these anaerobic gram-positives. Actually, you saw them also in the ileum, or not in the ileum, in the colon. Those are also absent in the ileum so much, or low level. They're also low level in the babies. But they are there, as you can see them. So when we do all this high throughput stuff, we can find them. Actually, we did deep metadomic sequencing and found in an unbiased way that all those microbial species are there in the baby. This is 100 days of the baby. And there's not only bifidobacteria there, but also strict anaerobes, which are indicated here. Uractale, Ubacterium hallei, Akkermansia, Veilonella, they're there on low level. They're just there. You find the genes of them. You find the signatures of them. They're there. And if you look at the metagenome, you can see that the healthy babies, the adults, they share a lot of species together. They're not exactly the same, but this is just to indicate that, in a way, everything is everywhere the environment selects. That's not rocket science. Dutch scientists have been, Bas Becking and Beyering have been claiming this already 150 years ago. But how does it work and why is it important? Well, it's important because it explains what we see. It also explains what is happening. And how does it work? I will give you some idea. Why is it important also is illustrated here. This is our immune system. It's quite different in utero as compared to postnatal development. Also here you can see all the microbes which are interacting and educating our immune system. And there's a co-development of our immune system and the microbial colonization. So here's where it all happens. If this goes wrong, it may have profound effects. Good to keep that in mind. This is not happening, well, in germ-free mice it's a bit different. So we have to be really modest there too. So this is different. There are reciprocal interactions between microbes and host. So how does it work? We did actually some proteome analysis. We didn't look only at the genome and the DNA, we also looked at the proteins. We have systems for that, we reported that, and actually, when you zoom in on the metabolic pathways of the bacteria, you see obvious things, that is sugars are being degraded, bifidobacteria are all there, they make this beautiful pathways, which includes phosphoketolase, they use the salicylic acid, the non-bifidobacteria actually have glycerol phosphate, triphosphate isomerase, which are key enzymes missing in the BIFs. You can see them all expressed in the other bacteria. Also, they're more involved in some of the, I would say, the fucose metabolism, which is an important factor. I will come back to that later. So this is all working. You can do that. But what we also saw is something special. The gut microbiome of babies is dominated by human proteins. In early life, this is meconium. Later on, you can see it goes down. It goes down in 4 years. We follow the same baby for 4 years of time. You can see it go 3 years. You can see it here. This is you and me. We took earlier individuals here. This is low here. So actually, early life is dominated by proteins, host proteins. They're made by the, they're coming in the meconium. They may be coming from the proteome of amnion fluid. We analyze that too. It's very similar to what the babies are swallowing in utero. So you see these proteins there. But later they make themselves. And also it comes, of course, from the mother because we know they deliver also the immunoglobulins and the antimicrobial proteins as well. But also the baby is making a lot of these proteins. And we zoomed in on one of these proteins. Actually, we call it salsa, but actually the official term is DMBT1. It's a deficient in malignant brain tumor. You don't want to say that all the time. So we, and this has maybe a negative connotation, it's just discovered the first there. We call it salsa. It's actually very abundant in your oral cavity too. It's a protein which agglutinates bacteria. It's also very abundant in amnion fluid. I will show you in terms of volume. This protein has been analyzed. It's a dual function. It agglutinates bacteria to each other. It also triggers the immune system. It interacts with the complement. So early life is a program to balance outgrowth. So why is that? I think this kind of proteins play a role. What's our evidence for that? Here you can see a couple of babies we followed up in time. This is now online. Amnion fluid has this salsa protein. This is both the readouts on proteome, as also is an antibody. And here's the meconium. You can see meconium is full of it. It goes down a bit later, but it's still there also after 1 week. This is, by the way, with different babies. So they have a variable amount. This seems not so high, but if you calculate, there's a half a liter of amnion fluid there, whereas this meconium is only a few grams. So there's a massive amount of this protein present in the amnion fluid. What we then did is took this protein and analyzed it for binding to bacteria. We find it binds pathogens. This was a bit screwed up in the plane when I made this. I put this there. But then basically it comes down to the fact this is supernatant of this salsa with a protein, with a bacterium, and this is the eluted one. So this is supernatant, means that's all there in the supernatant. It can't be eluted because there's nothing there. However, here you have the supernatant here, and it all can be eluted, meaning that the salsa is binding to the bacteria. So we have pathogens that bind, and commensals, some bind and some do not bind. And I think that makes a difference. This actually explains why this kind of antimicrobial proteins on a structural way may also shape the environment in early life. Now, this is a picture you saw before. I was happy that Josef showed it because actually it's a picture from my lab. It's made by Nora Ottman, and she's a PhD student. And she's artistic together with Clara Bell, so she made this nice picture, and it zooms around on the web. That's why I usually don't put all my slides on the web, because you have to put it in the context. What the context is here is the development in early life and later life. I'm not going to talk about old life, but we had dividend work with Claudio Franceschi in Bologna with centenarians, and they have signs of inflammaging, cytokines in the blood, reduced microbial diversity, and reduced butyrate producers and more beta vions. But you see the same signatures. You see also in earlier life in crying babies. We address colic babies. And actually we followed that in time very nicely, 10 samples for 100 days, several babies, quite a few, hundreds of analyses, and we found that we could see already early warning signals of this crying before they started really crying after 6 weeks, and we saw the same pictures here. That indicates that this whole thing has a very important impact on the whole immune system. Signaling and immune impact are evident, as I indicated to you, but also early biomarkers can be developed. Coming back to the story of the early life, we have to be aware of all kinds of confounding effects. And one is the use of antibiotics. The Blazer group has shown that. This is Marty saying stop killing of the beneficial bacteria. Well, we can disagree maybe on that, but then it depends how you see it. Of course, antibiotics are very useful, have brought us at the stage we are now, but then too much is too much. If you have that all the time in mice, you get a different microbiota, but it has been done already in the early 50s. This is a paper showing that the growth response of rural Africans, South Americans, is affected by antibiotic use. There were not all the modern antibiotics there, but they looked at that. And actually, if you look at the paper, it says whether the antibiotics have an effect on recovery beyond their influence on infection, because these kids have all kinds of infections, is still uncertain. And therefore it works. But in analogy with studies in animals, production animals, don't forget that, it would appear that there may be also favorable effects on their intestinal flora. It's good to keep that in mind. So it's already predicted 50 years ago. Now, how does it work? We actually addressed that in a very coherent way in longitudinal analysis in Finnish kids. Finnish kids go to daycare. There's a highly educated society in Finland. I'm now there 7 years. High level of education, 80% of the women are employed outside. They all go to daycare. I can't have meetings at 4 or 5 o'clock anymore because my postdocs have to get the kids from school, from the daycare. You know, they have to go there. 5 o'clock, they have to be there. So my meetings are at 2 o'clock because, you know, I can't get them later. They have to go, otherwise they can even find nothing when they go to the daycare. So all the kids go to the daycare. It's free, like healthcare is free. Everything is free in Finland. Education is free. It makes life easy. Not only as a foreigner or a European citizen, but also for studies like this. Because all the data have been collected, because everything is reimbursed. So you can see what kind of medicine they have had. So we looked at this kids cohort, and we got permission to look at the records for about 60–70% of them. And we looked at 88 kids, and we found they use a lot of antibiotics. Because they're in the daycare all the time, they have ear infections, and they infect each other maybe, and the mothers want to work, so they put to school with a bit of antibiotics. But there was a special cohort in the north of Finland, in Oulu, and actually here you can see the purchase of the antibiotics and the recall of the parents. I got first the data of the recall and thought, well, we have to go for the purchase too. Here you can see the difference, but it's very similar as you can see here. But we're talking about 25 courses of antibiotics in the same kit. This is what the data show, purchase data, recall data. So I believe that. So that's how it is. So we started to look at them. We look at the microbiome, and we also looked at BMI development. We got this data and the permission to use all the data. Here you can see courses of antibiotics, BMI increase. This is 4 years, 5 years, and 6 years old kids. Why did I separate them? Because the diversity is still changing, as I showed you. So we segmented them into years. You can also put them together. It's the same thing, but it's nicer here. 4 years goes up, 5 years goes up, 6 years even more, because you have had more courses you can, and more effect to implement. BMI is related. You can calculate it's about 1 kilogram per course of antibiotics. So this is correlation. Is it causation? We can talk about that, but I think it's pretty strong. Then we looked at the microbiome analysis. So we look at the metagenome analysis, and actually we looked also at the colonies. We looked at the link between macrolides, for instance, here, and ear-to-resistant colonies. There's a nice correlation. Well, it's not rocket science either. We think it's causation. You give macrolides and you get macrolide resistant. So I think this is causation. The same as the genes. They also come up. Takes a bit longer because probably they're not all the gene transfers are there. You get the first reaction of the microbes are resistant. After a while, you can see the resistance genes spreading in the community. So we see this antibiotic resistance tenfold increased after antibiotic treatment. Is it causal? I think so, but still this is an associative study. It's a longitudinal study. Sugar transport, other metabolic properties are changing. But we also found a 4 times increase in bile salt hydrolase, genes which are important in the bile cycle. I'm not going to talk about that. I think other people will do that. But fat is an important driver of your microbiome community and actually signals all the time to the host. You don't want to eat so much fat. That's a good thing, because if you do that, and Peter Turnbauer showed that, you get a real difference in your microbiome. However, we're not going to eat 70% of our diet in fat like in those studies, so we have to be careful there. But here you can see reduction of bile salt hydrolase. Now we know from work actually done in APC in Cork with Fergus Shanahan and other people there, that if you manipulate the bile salt hydrolase levels in an intelligent way, more by age is less obesity and high-fat diet in mice. Not sure whether you can translate that immediately, but it tells you something that this may be an important finding. So here we are with this cohort. We took it a bit further because we followed the kids longer in time. Then we found out they were taking asthma medicine. And we interviewed them. They are having asthma. Some of them, not all. The ones that have asthma are the red ones here. Those are the ones which have eaten the macrolides. You can see the more macrolides, the more asthma. And actually you can look at the microbiome too. And you can take them and make a nice IRO on the curve and a prediction. A very nice prediction of your asthma in your life. Based on this, this was 2 years before the asthma development. So actually we could predict that. Of course, it has to hold up in larger cohorts, but we work on that. Microbiome can be used as an early biomarker, and we have now a large cohort that we follow up now for 12 years to look what's happening there. So these kind of things are important. So this is almost coming to the end. I have another 15 minutes maybe to finish off. Am I speaking too fast? So this is to illustrate something else. You know, our microbes are not alone there. We have genes too. We have also quite a lot of genes, maybe a bit less genes, but they may be more functional. Otherwise, we are not microbes. So we have to be also aware of that. So we have this interaction between the genomes. Well, are they really interacting? Well, there's only a few data showing that. Josef showed something about the ACE protein, but that was in mice. So what about human? Can we see something of that? Well, we're not there yet. There are millions of SNPs there and there's zillions of SNPs there. I will show you also later how that works in the GI tract. So we looked at a special phenotype. That's the secretor phenotype, and you may know it. Non-secretors have a SNP here, and they don't make fucose on their sugars. So basically, they're not fucosylation. And secretors have that. There are 2 genotypes. You can see it separated a little bit. I think it has to do with the efficiency of fucosylation. But you can see the microbiome is changing too. We find a correlation between the non-secretors. There are organisms like Dorea, Cannabis, Baudia. This is a homocitogen, and actually linked also to the enterotype 1, and that's more profoundly associated with IBD and IBS. Actually, also non-secretors are related to Crohn's disease. So you see microbes also here. For those who are interested, Trumulus canavis is one of the organisms you find always in IBS. It's an organism which we don't want. We now isolate it and sequence it. Here we have other organisms. These are present in the fucose producers. More bifidobacteria, more Akkermansia, and some Bacteroides species. All actually organisms that are fucose users. That's not rocket science either. You give them fucose, they come there. So they have a special interaction. Early life, they're colonizing early life, and the mucus colonizers. So that's the idea. And this fucose is not only on mucus, but also erythrocytes. Actually, the whole processing of the glycosylation is affected in these non-secretors. it has impact of all kinds of diseases. So this is the kind of relations you can think of. So early life, these are early life colonizers, that's important too, but also the genotypes plays a role. Of course, we want to look more further into that. We have the studies ongoing because a lot of Finns have been sequenced nowadays, so we can look at that also in family studies. I want to focus attention not so much on bifidobacteria, because maybe other people come back to that. Akkermansia is my favorite. It's an organism which we pulled out about more than 10 years ago, and actually it's a dedicated mucus utilizer. It was a new organism at that time. It's a microbiome, it's an anaerobe, 3 million base pair genes, a genome, all kinds of genes involved in mucus utilization. It's low in obesity people. It's low in IBD mucosa. It has a link with health. It's there in healthy people. It signals to the host. So what I've been doing, actually, was named after Anto Akkermans, one of my co-workers. When he retired, we usually name species after people when they retire. Rombautsia is named after Frans Rombaut. We have a couple of those around, so when you retire, you get your name there. The problem is you don't know whether it's a good guy or a bad guy which is named after you. So I said I don't want that. When I'm dead, it's fine. Okay, anyway, so this is this Akkermansia. This is sitting there in the mucus. It's a mucus colonizer. You can see it nicely covering the mucus layer, very nicely, like the A's in a way. But however, it's in the mucus layer. And you see here what it does. It makes mucinases, all kind of enzymes, and we all analyze them, and it cleaves the mucus into oligosaccharides, which is food for others. There's trophic chains we can make. We actually can grow Akkermansia with Faecalibacterium, and it makes butyrate and so on. So you can initiate trophic chains. Butyrate is produced via other bacteria, but it makes propionate and acetate, and that's actually all fueling the cells, but also signaling to the immune system and DPR receptors and toll-like receptors. So we have worked that out in detail, but I don't have time to talk about it. So this organism is of interest. And we did an experiment with Patrice Cani. This is the only mouse experiment I'm going to show, but I think it's quite cute. What we did is we thought, well, if this Akkermansia is so important, can we see something? And actually, he discovered in his work on mice that it was also depleted in high-fat and obese mice. So we took this normal mice and put them on a high-fat diet. Here you can see what happens with it. They get more LPS in the serum. Think about what I explained before. LPS produced by proteobacteria. They're mice, but still maybe the same mechanism apply. If you have a high-fat diet, you get damage. You get more proteobacteria making LPS in the blood. You give Akkermansia. You see, it goes down. You give killed Akkermansia, it doesn't go down so much. So you need live cells. Here you can see the fat mouse. Control mouse, high-fat mice. High-fat mice get fat. That's not rocket science. Give them Akkermansia, they go down. Killed Akkermansia doesn't do it. So this was repeated several times. I didn't believe it. You know, I'm not paid to believe, I always say, so I want to see the data. So we did it a couple of times with dead cells or whatever. I did it again. Then we published it, it came into Nature, and it says obese mice lost weight. It's not the case. There's not obese mice. Yeah, well, they're obese, but we have to have a high-fat diet. So we changed something. And this is also what I want to warn you about. I mean, don't believe everything what's in Nature. I mean, it looks nice, it's not completely correct. So look at the facts. The same is this what was it, this artificial sweeteners. If you drink 6 liters of cola, yeah, then you have a problem. But you look at the doses, and there was only 6 people, you know, of the 10 or 4 of the 10 reacting. And Max knows it much better than I do. We were interviewed about it. I didn't want to comment on that because I think it was a shitty paper, to be honest. I like the idea, but it's not good. And you have to look at that. So here, fortunately, this was also reproduced by a Korean group. They also took Akkermansia. They put it into high-fat mice without metformin because it was about metabolic syndrome. And you can see they actually attenuate adipose tissue inflammation by inducing their Tregs. So here you can see the evidence. In mice, it goes to Tregs. So this is the model we have for it. And actually, you may have seen that. It was alluded to it already. This short-chain fatty acid, they signal to our DPR receptors. Actually, they signal to Tregs. And this is a sort of summary of work of Wendy Garrett's group where they looked at this signaling. And you can see here the summary of Fiona Power. You can see here fibers in the colon will make then this sort of acids, and they go to the Treg cells. And, you know, feed your Tregs more fiber. I like that. You know, it's very, everybody can understand that. I mean, this is fiber, the fiber hypothesis. I will say, make more mucus, or put Akkermansia there. Because Akkermansia stimulates mucus production. We have seen that in mice, we have seen it in human. Globule cells are activated for mechanisms we are working out. More mucus is produced. It has an autocatalytic runaway situation. It promotes itself and then makes more propionate, it makes propionate, a bit of acetate. And also with trophic chains even make butyrate. So this is what goes on. So it has this cycle there too. We show that in germ-free animals. We look at gene expression already years ago. We also use this mice organoids, like I showed already from the Clever's lab, and we showed also the signaling of Akkermansia and Faecalibacterium. Akkermansia signaling much more than Faecalibacterium in those systems. Human trials are ongoing for that. Back to the diet, because I think I finish off with the diet. We know that periodization overrides fluctuation normal diet. However, we can manipulate the microbiome with diet. We can do that. If you give a high-fat diet, it's even more, but with this very low-cal diet and this prebiotics, you can see effects. This is what happens. This is work of Harry Flint, and we worked with him and analyzed this data again. Here you can see this is a maintenance diet of about a dozen or so type 2 diabetes males. You give them non-starch polymers, wheat-bound starch. You can see not much is happening, but here you can see the things are happening. Give them starch. Give them just there is a starch that goes to the colon. The fibers are there, and you see a major change, a bloom of two organisms. You can look at them, the gold ones and the red ones, Eubacterium rectale, Ruminococcus bromii. They're interesting organisms. And actually, we did some work on that before they published this already. With Petya Kovacs, as a postdoc in Fred's lab, we used C13 starch and put it to an artificial bioreactor with microbiome, seeded with a colonic microbiota. And we saw there was a trophic chain being formed. Basically, Ruminococcus bromii makes amylase, makes acetate, and Eubacterium rectale takes it up. And you can see that in the C30 label going there. It comes in C30 butyrate. So we could see that all with the metabolomics. So you can see that. So these trophic chains are there, and we can show them in vitro, and I'm sure we can do them also in vivo, and we work along those lines. But it's also important here that we actually use this data. I'm not going to in detail, others will do that, I think, to do microbiome-based predictions of responders and non-responders, and associated to the type 2 diabetes intervention, as well as the insulin resistance. So that's very important, and we actually expanded that to other data sets. We didn't only use this data, we used actually data we did with Nathalie Dalzell and Patrice Carny and with other people, also in Finland, and we put all this data together. And we still could predict responders, non-responders. So it seems to be a very robust system based on diversity and specific microbes. Going back to this cycle, I tried to convince you this is true. Well, I have to say also, this is a model. A model is a hypothesis. And a good model is a good hypothesis. And that's how it is. That's what I teach my students, and it's good how it is. It gives you a shape of mind. It is not proof. Even when there's a good model, it's not a proof. It's maybe, you can assume it, but it's not proof. So we are here, and I give you some, I would say, evidence for it that it may be like that. But the best evidence comes from the microbial restoration. This is where the promise of poop comes. I mean, this is from Max. He was interviewed in Science. I mean, you have this Dutch science writer, he writes about it. So we did this work on the fecal transplantation in C. diff patients. There's a long history. Traditional Chinese medicine. I haven't seen Lippen Zhao yet, but then maybe he comes back to it. Is he already here? No. Oh, yeah, here. Hi, Lippen. You know this. I mean, that's, you know much more than I do. It has been called the yellow, what is it, yellow sauce or so, yellow soup, I think it was called. And many single cases have been reported, quite successfully. But we did a study actually with Josef Keller, who was there sticking out his neck, but also with Max and other people, else van note, and actually we found that in this case, bugs are better than drugs. We had a vancomycin treatment, we have a lavage, and we had a lavage and transplantation. The first two didn't work. Actually, the medical ethical board told us to stop that intervention. It was not ethical anymore to don't give them the microbes. So the microbial fecal transplantation was superior. So people are treated like that. When you look at what happens there, you see spectacular effects. The donor has this kind of networks of microbial communities, these co-occurrence networks. They're not there in the C. diff patients, but you restore them in the healthy situation. Actually, there was a paper also, I think, online. I think I was just informed this morning in the bus by Wim Saris that there was a paper out in Nature also addressing this in mice and take this stuff there. But it's fine. The proof of the pudding is the eating in human. It's good to go there. So how do we do this? Actually, we do it with Max. It's nice. I like this thing he has here. This is the transplantation. We did another experiment he will show about it, but I want to show you actually what we learned from these transplantations. This is an experiment where we did an autologous transplantation. That means people go in there and get in a double-blind way their own microbiota back, autologous, or they get a healthy donor. So this is all, well, I would say, quite sophisticated. You can't do it better. So we did that. So we took this data set because you can't do it better. We looked at the metagenome and the SNPs in the donor versus recipient, together with Pierre Borg, because this is a computational, quite challenging experience. Here you can see the autologous transplantation. You see nothing is happening there. Everything is 100% the same in SNPs as a donor. Yeah, no rocket science. We could take them out immediately because they are the autologous ones. Those are the ones which have the healthy donor transplantation. They get better. Max will talk about that. You can see all kinds of things are happening here. All kinds of SNPs of the donor microbiome are established in the microbiome of the recipient. This is after 90 days, 3 months. Actually, when you look at that nicely, you can see in the gradient going. We took the 2 days, 14 days, 42 days, 84 days. You can see it's all going there. So some bacteria species, which are coming from the donor, are establishing themselves. You see the SNPs back. Their signatures are there. So some lose and some win. And why is that important? It's important to learn from it, and also it may impact the synthetic microbiome development, how to take those things out and take the most competitive strains and so on. So it has an impact there. So this kind of work we're doing to understand more about fecal transplantation. So this is what I say is a discovery tool, because it's a fantastic tool. First of all, you cure people. Second, you can learn from it. And third, I think it's therapeutically applicable in the future. So that's just why we do this. So we do this reverse engineering. We select the right microbes and actually expose also the relevance of the ileum there. And we do reverse engineering, and if it doesn't work, we go back. We do mice models of proof of concept. If it doesn't work, we can go back. Large-scale production, human trials. This is where we have to go. And this is the avenues we take, and the fact that we have cultured already quite a few organisms. We have now a new institute funded by the government. You don't get a lot of money from the government in the Netherlands, unfortunately, but we were so happy to get funding, 25 million, for a new anaerobic microbiology institute, actually spread in different places in the Netherlands, named after the first professor of microbiology in Wageningen, so I feel very happy with that. And we have this fecal transplantation we work on. So actually, that is where we're going. I think it's time for coffee. What happens now here? We have this, I think, this impact of radical innovation. This is different from what markets indicated. I think we're on the doorstep of real changes. Why is that? Microbial diagnostics is evolving. And we can use that to differentiate responders, non-responders. That will come back later. And microbial therapies are there. So we can exploit this microbiome. We can stratify responders, predicting responses. We can dissect the mechanisms, exploit the targets, whether it's going to be in mice or in human, I don't care. Whether it works, it works. It's a large promise, of course. And we have to see how it goes, but they grow together. Both food and pharma are going together. I'm happy to see also people in the pharma industry here, because this is where we go. And that's where different niches can be instrumental in developing this area. What's next? Lesson from large data sets. I think that's what's happening. Why is that important? Because you and me, we're different. So we have to unbias the bias. We need validated technologies. We can have a whole series of lectures about that. I'm not going to do that. Longitudinal studies are useful too. Follow things in time. Look at early warning signals. I'll give you examples of that. Personalized approaches, personalized nutrition or whatever, precision nutrition, whatever you can think of are there. New insights from certification there. There are a lot of diagnostic opportunities. And I can't emphasize more the impact of early life colonization on later life health. I think we have something here which is really important, and with the premature births and especially the cesarean deliveries and the whole hygiene changes in our society, use of antibiotics, other food, we have to address that. And I think it's a matter of public health as well as opportunities for companies to go into that. Fecal transplantation is a discovery tool. I indicated why that is the case. We can learn more from the host microbe interactions. And I think we have the proof of the pudding is the eating. Bugs are better than drugs. Well, show it. Show it and show it in human, not only in mice. Show it in human with either a nutritional or a pharma intervention, and show it works. I think that's where we are, because we can have a lot of promises, but we discussed it last night with Dusko. We got a little bit more gray hair, so I want to see the results. So therefore I also say I don't work so much with mice, because they're so nice. And you can do so wonderful things, but you will never come back to the real life situation. You have to go there in order to drive the innovation cycle. So if you want to have advice, go there and see what you can do, because that's where the action is. Thank you very much, and time for coffee.
So maybe there's time for 2-3 questions.
Maybe I take one, huh?
Yeah, please.
And thank you very much. Indeed, I thought Italians spoke quickly.
Yes, I give the disclaimer already. It's like when you go to a pharma conference, you have to have your disclaimers with all the companies.
Everything is everywhere. Now, maybe you said it and I missed it, but I'm really intrigued by this equilibrium situation. So what are the factors that triggered the return to the initial situation? And are they understood? What's happening there?
Yeah, I think that a good question. I think we have this balls, this equilibrium. Changing environment may change this bistable organism to change, and they get a domino effect. I didn't expand on that, but that's how it is, at least in theory, and experimentally addressed in some of these systems that have been published. What do we know about the gut? I think antibiotics are such a driver, and they may change this system. And then you go into an equilibrium situation which is different from before, and then a little change can make it flip. And that may explain also this asthma developed later on, or it was there already in the first place, we do not know, but then this is happening. So little changes then may affect it if it's already changed. So antibiotics, maybe cesarean delivery and not the right microbial colonization, maybe priming the immune system in different ways. I think that may also predispose for this alternative stable states. And I think that's the explanation. However, we don't have real evidence for that yet, and I think longitudinal analysis, together with real profound and deep analysis of the microbiome as well as other host factors, are needed to look at the associations there and maybe then also come to ideas about causality. Thank you very much.
Here's the next question.
Yes, Willem, thank you very much for this excellent talk. So most of the human data are on fecal analysis, so meaning this is a kind of limited resolution of what is really going inside the human intestine, in particular the upper gut. Can you give any kind of indications how much it tells?
Well, in numbers, I indicated to you, it's about 1 million to 10,000 fold less in numbers. But we can see, for instance, intestinal bowel overgrowth in some of in the fecal samples. We can detect that, and we can see streptococci in the fecal samples. So they come out at the end of the day. If there is many, they come out more. So it's a matter of depth of the analysis. Therefore, I think it's so beautiful that our metagenome and other tools are developing that we can go deeper and deeper with our molecular microscope. So what has been there comes out. That's the idea. Of course, it's not always like that. There may lies and DNA may be damaged and eaten up and whatever. But still, I think that's the idea. We look also on the proteome level, because with proteins, you can have a very sensitive analysis. I think about the ELISAs and so on. So we use that too to actually try to see what happened in the upper GI tract, because that's where the action is, as you indicate. And it's not so easy to sample all kind of people all the time and go there in with a tube. So we do that with a selected group of volunteers, and we use this ileostoma patients to do dietary interventions, because you can come back after a while and take samples. However, that's not for normal people. So we have to be smart enough to get there. But I always tell, if we can get people on the moon, why can't we get some samples from the GI tract somewhere? You know, we have to be smart enough. So please, you take the challenge.
So here's the next question.
One short question with regards to the factors that influence the early development. You alluded very, very clearly to the vaginal delivery versus sterile delivery as one of the early inocula. And then, of course, to the feeding and the substrates that bacteria get. What about the bacteria babies get if they're breastfed through breast milk? Can you comment on the degree to which that can act as a modulator?
Yeah, I like the question. It alludes to the fact that, you know, it's not only a fecal transplantation, everything is everywhere. However, the microbes you find later in your gut are anaerobic microbes adapted to the gut. You don't find them in the vagina. You don't find Faecalibacterium or Akkermansia so much on the breast milk either. However, they have to be there, but they can be fed. And of course, there's also fecal breast connections. I mean, everything is everywhere. And I think Rob, I'm not sure where he is, he has done beautiful work on this aura of microbes you have around you. So, I mean, you know, everything is there. And so it's a matter of anecdotal evidence. You can look at the breast milk or the breast is there, but it may be somewhere else too. So it's not so important where it is. It's more the fact it is there and it will come. But the microbes that we get in our early life and also later life, your microbes are anaerobic microbes that come from your family members. They don't come from the tablecloth. However, in premature babies, you can see signatures, and so Rob has been instrumental in that, signatures of the tablecloth in the hospital or whatever of the nurses or so. I mean, you can see other things there. Mobile phones are maybe an important source.
So maybe I have two final questions, Susanne, and then the last question after.
You said that young children showed that young children share the microbiome with their mother. They build it up. Can you distinguish between mother or caretaker?
Yeah, good question. I like to distinguish between mother and father. You know, I have been doing a lot with my kids, but okay, still, that's a good question. I think we could. We have a set of experiments ongoing in several hospitals in the Scandinavian countries where they have premature babies. They either kangaroo them, so they have them very close to their family members, and we monitor exactly what's happening there, as compared to hospitals where they don't do that. And I try to figure out what's happening there. It's not so easy, but I think it's an important question to ask.
So here the final question, please.
So related to the previous question, in terms of program microbiota transportation between mother and infant, I was wondering what is the influence from father?
The influence, sorry, is the?
From father?
The father, yes, I alluded to that. I don't know about the father. I would love to have it. Like I said, so in that experiment the trial we're doing, we sampled the father too. But in most of the data sets I could get my hands on, there's no samples of the father. So we have the term and so on, and Fred has been working on that too, and the term 1, term 3, and so on, the different parts of the pregnancy and the programming there that you can get also from the same cohort, maybe in Turku. But then there's not many samples available. So we're setting up trials, and they're running for a year now.
So the ultimate question from Stefan.
And then just before the coffee, I would like to go back to this equilibrium. Is there a time when, if you were to take a picture of our microbiome, that represents like a memory stage, and whatever flip, we tend to go back to that memory stage? So is there a memory of our microbiome?
That's a good question. I think we have done some work on the longitudinal analysis for over 10 years. Some people in the audience think it's good to freeze the microbiota and use them later for transplantations. I also froze my microbiota too, but only at the biopsies, I think, not the real thing. Is there a possibility there? And I think there may be. There may be a sort of window of opportunities that we say, well, this is where the microbiota is stable, you're healthy, in maybe whatever part of your life, keep it and use it for later. That's all the challenges for the elder life, and maybe Paul will come back to that in his presentation when he talks about the elderly. Thank you for the question.
So thank you again for the lecture. And I propose to reconvene around 11. You're the second. I think you can set it up here. Somebody will help you then. I think you just plug it in. You want to? I think you're the second speaker, right? So you could just.
It's coming.
Ah, it's coming.
Okay,
Great.
perfect.
Okay.
Good. Thank you.
Yes,
of course.
Yes.
Should be fine.
Should be fine, but just in case.
It's always better.
Yes. Just setting up here, or?
He's coming, he will...
Okay, maybe I should wait. Sure. Oh, he's up there.
Yeah.
Maybe I can try this.
I'm not a specialist, so... I don't think so.
So maybe not. There you go. It works.
So it's okay?
Mm-hmm. Yeah.
Okay, so it's fine. Okay.
Yeah.
So it's perfect.
Perfect.
So you know how to do it.
Yeah, I know how to
afterwards.
do it. Yes.
Thank you very much.
Thank you. Thank you for your help.
You're welcome.
Il en faut combien? Une? Il y en a une là-bas à 1 minute. Lui, il utilise pour sa caméra. Ouais.
Je monte avec toi pour mettre la vraie.
On va l'installer ici.
Ah. Et je voudrais, tu peux couper les écrans 2 minutes, je voudrais
coupé
changer. Je veux changer à...
Ouais, j'arrive. C'est coupé déjà. Numéro 6.
Et à midi, je suis
Hey, bonjour. Hey, it's so good to see you.
It's been a long time.
Yes, too long.
Yeah.
Too long.
Everything's good?
Yes. How about you?
Yeah, well,
It's
busy but nice.
Yeah. When did you come?
Well, this morning.
This month.
Just a few hours ago. Just had a
You
shower.
are a strong man.
I'm
so.
So, but anyway, we should talk.
Yes, sure. And
I have lots of ideas.
Good. I'm sure you have. You always did.
Tu peux y aller pendant ce temps, t'inquiète pas. Moi, je vais lancer.
Tu t'occupes de monsieur Dusko? C'est vraiment le prochain speaker.
Voilà. Donc, j'ai connecté ça. J'imagine qu'il faut connecter ça.
Oui, je m'en occupe.
Après, on va essayer. Voilà.
Okay. Do you use a sound? Yeah?
No.
No. Opposite.
On doit déplacer un tout petit peu pour que ça tienne. Parce que bon, ils font plus le...
C'est dommage. Oui, just. Compliqué. Allez.
Comment? Ça, oui, ça va. Oui. Ça va. Oui.
Cool. Ça tient. Je vais vous switcher sur le screen pour qu'on puisse voir si ça marche.
Ok, très bien. L'adrénaline, ça...
Non, c'est ils ont des secondaires. Heureusement, tu parles un peu plus lentement quand même.
Un peu.
Parce que sinon, ça serait épuisant, c'est tout le concret comme ça.
Ouais.
Tu veux un café, Dusko? Tu veux que
C'est déjà fait, c'est déjà fait.
So welcome back, everybody, after the break. Oops.
Try again.
So welcome back, everybody. I hope you. So after you did a glucose tolerance test outside and fed your microbiome, so I think we had a great beginning. This field is ripe for lots of discussions, lots of interesting studies published. I think we need some critical discussion and discourse. So I think we had a great beginning, and now it's a pleasure to introduce Dusko Ehrlich. So Dusko was trained in organic chemistry at the University of Zagreb in Croatia and got his PhD in biochemistry at the University in Paris 7. He then worked with Joshua Lederberg, a Nobel Prize winner at Stanford University, and he founded and directed the Microbial Genetics Research Unit and the Microbiology Department at the National Institute for Agricultural Research. And so he was the founder, currently is professor at King's College in London and chief scientific officer of a company called Enterome, which tries to develop microbiome-based biomarkers, which of course of interest to many of us. And he has made major contributions in understanding the human microbiome, metabiomes, and of course the definition what's a healthy human microbiome. And thank you so much for coming, and we're looking forward to your lecture.
Thank you very much for the introduction and for the invitation. It's great to be here and talk to you. You'll see some of the themes that Willem so wonderfully raised will be reoccurring in my talk. But to start, I really like to show a slide which reminds us that, that one can think about human intestinal microbiota as a neglected organ, large mass, many cells, and major role in health and disease. And I illustrate that for chronic diseases with this slide, which goes from frailty in seniors all the way to cardiovascular disease and many others. Too many diseases. So the slide would get longer and longer, so I just stop it here. An important thing to remember also is that chronic diseases are steadily increasing in industrialized countries, and there is no prevention and no cure. So it is a major health problem. Of course, prevention of these chronic diseases could impact public health greatly. And a simple way of viewing prevention is saying, well, there are two components, risk detection and risk alleviation. It's not enough to detect the risk. You also have to do something about it. And we just wondered, could the microbiome inform on the risk of chronic disease? Can it be a target for intervention? I'm sure that you recognize some of the themes that Willem raised. But we felt that we are lacking really tools to assess the microbiome well enough. How to assess the state of this neglected organ in each and every individual? And via MetaHit Consortium, we raised to that challenge by saying, okay, we should probably start by describing what's the status of what got to be known the other human genome, what collectively we can carry in our intestinal tract, and then try to find out what is the other genome of each and every individual. And for that, we developed the approach that we name quantitative metagenomics. So what is quantitative metagenomics? Quantitative metagenomics, we start from stool samples and go all the way to predictive models and the indication of how to go about it. And how do we do that? We have developed methods which make it simple to collect stool samples. That can now be done at patients' homes. We have developed quenching liquids, so composition of microbiome is not altered for a week. These samples can then be sent by mail from the patient's homes to the lab. And when the lab receives it, then all DNA is extracted from that. And that captures, of course, all the genes that the individual carries. To find out what are the genes in there, we sequence a lot, generate many, many sequences, 30, 50 million sequences, which capture presence and abundance of the genes. They're short sequences, and they're not informative on their own. So we basically use them to map on the reference gene catalog, which contains all the genes that we have ever found. Map, and whenever we map non-ambiguously short sequence to this reference gene catalog, we count the genes. So we get gene profiles for many individuals, millions of genes, and then we relate these profiles to human data, and from that we identify relevant microbial players, build and test prediction models. And in principle, it looks quite simple, and it is, but there is a critical point. This needs to be standardized, because if, say, collection is not, or storage or sample processing is not done quite right, then it's very difficult to correlate things between different studies, and that has, I think, impacted the field to some degree. Now, the most current reference gene catalog is the catalog of 10 million genes that has just been published a few months ago with a big contribution from BGI. It's great to have Ang Jun with us here. Maybe he'll expand on that later on. About 1,200 individuals that were studied. And what is one of the messages from here? Remember, the first catalog we published that got this Nature cover that was shown was on 124 individuals, 10 times more, and contained 3 million genes. Now, one of the main messages is, well, the genes that are common to many individuals actually don't increase very much any longer. So it's like common variants. Genes that are present in about at least 5% individuals, well, there may be between 3 and 4 millions of these. We have seen them. Now, what's intriguing, of course, is that rare genes present in very few individuals are increasing. Maybe they contribute to that notion that we are also different. But maybe also part of it is that some of the microbes that live in our body don't really reside there, but rather transit in there. Be it as it may, we think that these common genes will be most informative for common diseases, chronic diseases that we would like to tackle. But beyond 10 million genes, we also wanted to cluster the genes in classes of genomes they come from. And we did that. I won't go into details of the procedure. It really goes to looking at the abundance of genes in different individuals. Genes which have same abundance across different individuals belong to the same species. What have we found? We have found, if we look at the frequency of objects, clusters that we have formed, which we call metagenomic units, here that some are large, have many genes, and they really represent species, bacterial species, which we call metagenomic species. We have described, it's also a paper that came out earlier this year. We call these metagenomic species. 85% of them were previously unknown. So when only the known species, reference genomes, are being used to characterize microbiome, one is characterizing only 15% of all that is in there. But there are not only these large species elements, there are also smaller units which have, say, less than about 700 genes. Maybe it's mean is around maybe 50 genes, and they correspond to phages, viruses, plasmids, CRISPR elements. So we start having a really detailed description of this whole microbiome. We can construct these interaction networks. You can't have a phage if there is no bacterium on which the phage feeds. So we know about these relations, and this is a zoom on one part of this network. This particular metagenomic species, we see some phages which will attack it. We see an immunity element. And when this immunity element is present in some strains but not in the others, then this phage will not attack it. So we do have a detailed representation of what is going on, and we can characterize our neglected organ. We can assess microbiome and each and every individual to an unprecedented detail. And you can even think about this quantitative metagenomics approach as a powerful microscope to scan the neglected organ, ways away from Antony van Leeuwenhoek's first microscope some time ago. Yet another Dutch person. So what can we then do? I'll illustrate what we can do in the following slides, giving very few examples. We can do diagnostics. A paper we published a couple of months ago on liver cirrhosis, done in collaboration with our partners at Zhejiang University in Hangzhou. A cohort of about, well, almost 250 people, patients, liver cirrhosis diagnosed in different ways, controls, people who come for health check. We split our cohort in a discovery to find out species that differentiate. I'll show them in a few moments, patients and healthy individuals, and train the algorithm, which this diagnoses with about 94% accuracy between patients and controls. And then we used the remainder of a cohort, which was never ever used, to train the algorithm, and we get exactly the same type of precision. With only seven species, we have this accuracy of diagnostics. So many times people ask, well, what are the confounders? We have eliminated as confounder a disease status. We can as accurately diagnose compensated patients and decompensated patients. We have also eliminated the effect of medication, antivirals, beta blockers, proton pump inhibitors, because we can distinguish, diagnose people irrespective whether they take these drugs or not. So today, golden standard for liver cirrhosis diagnosis is biopsy. And biopsy is an invasive method. And you can't do it. It's not simple. We are replacing with a non-invasive method, stool analysis. So we can diagnose with high accuracy. But we can also say something about, beyond the diagnostics, we can say something about the status of a patient. I'll take a few moments to guide you through this colorful slide. So first, we have organized our cohort. This is a discovery part, this is a validation part, into healthy individuals and liver cirrhosis patients. And then we represent 28 species, metagenomic species, that are enriched in liver cirrhosis, and 38 metagenomic species that are enriched in healthy individuals. By these boxes, each of the boxes really represents 50 genes of a species, and there is a color code for this representation. So each column here is one individual, 98, 83, and so on, and each line, each row is a gene, and 50 genes are displayed. So you can easily see that these 28 species are present mostly in liver cirrhosis patients, both in the discovery and the validation cohort, while these species are lost from the patients. They are not there. They are typical for healthy individuals. There are 38 of them. Now, we have also ordered liver cirrhosis patients here in terms of the abundance of these species foreign invaders. And what do we see when we compare the quartiles, say, patients which have least of these species and patients which have most of these species, that the clinical scores are much worse for people who have high load of liver cirrhosis species than for people who have low load of that. So we can not only diagnose patients, we can also say how sick they are. So what's going on there? There's a massive change of microbiome in liver cirrhosis. One parameter which I'll touch upon more later, they have lost very significantly richness of their microbiome, both in discovery cohort. These are healthy and patients. These foreign species that I've shown you, in some patients represent up to 40% of the microbiome. It's not a small change. It's a huge change. Well, in healthy individuals, we don't find them. But look, there are a couple of people here among healthy who have increased number of these invaders. And I think they should go back and be re-diagnosed because maybe they are actually sick. Now, when we look at the species that were lost from the patients which are enriched in healthy individuals, they are typical species that we find in the gut. For most of them, we don't have taxonomic or niche assignment. For those that we have, well, they come from the gut. But the species that we find in the patients, great majority is actually oral species. There's an invasion, translocation of the microbiome from the mouth to the gut. So we live normally with 10 milligrams of bacteria in our mouth. It's not good for you to have 100 or 200 grams in your gut. And when we analyze their biochemical, metabolic potential, we do see that they would impact ammoniac, raised, manganese raised, GABA raised, and these are the three factors that impact hepatic encephalopathy. So there you have it. It's kind of toxic microbiome. And it's interesting that in the literature you can find association of dental health and higher MELD scores, which are used to evaluate status of the liver cirrhosis. Okay, we can diagnose. We can say something about patients. How about trying to detect a risk at which these patients are? A little story that we published last year, high and low gene count people. Some people have low gene count. Some people have high gene count. They happen to be distributed unequally among the enterotypes. Low gene count individuals are mostly Bacteroides enterotype, high gene count, mostly Ruminococcus enterotype. If one looks at the species that they have, and here genes are ordered, individuals are ordered by their gene number, low gene count individuals accumulate species which are known or suspected to be pro-inflammatory, while high gene count individuals tend to accumulate species which are typically anti-inflammatory. And of course, there's a big dysbiosis, big difference in unknown metagenomic species, which are, of course, a majority. But, of course, which was absolutely striking is that low gene count individuals, and it's 1 person in 4. 1 person in 4. 1, 2, 3, 4. At risk. they have less healthy metabolic and inflammatory traits. And this gives some of the details about it, but what it really says is that low gene count individuals have increased adiposity, increased insulin resistance, dyslipidemia, and inflammation. And we have not done longitudinal studies to say, aha, this is a risk. But we talk to clinicians, and clinicians tell us that they know from clinical studies that people who display these phenotypes are at higher risk for type 2 diabetes, cardiovascular disease, hepatic problems, and cancer. Not only the clinical picture of these people says that they are at higher risk, but also over time, over the past 9 years where we had records, low gene count obese people have put on significantly more weight than high gene count people. So not only they're worse off, they're getting more and more into that direction. So we analyzed their microbiome and found that people who have high levels of certain metagenomic species gain less weight over time. And that's displayed here, BMI change. They gain much more when they have low level of these species than high level of these species. Could these have a protective role? We don't know what they are. They don't have taxonomic assignment, but we can place them kind of among butyrate producers, and we just wonder whether that's not one of the important facets of that phenotype. Now, that was observed in Danes, and we went and looked at French, and we find basically the same type of change, this bimodal distribution of individuals, high gene count, low gene count, with dysbiosis similar to that in Denmark. The cohort was lower, smaller, so the power was lower. So we found only 18 species, but 14 out of these 18 were also highly biased in the Danish study. We trained our algorithm to distinguish low and high gene count people on 49 French. 6 metagenomic species could identify French with 95% accuracy. And the same algorithm, very same algorithm, works as well for Danes. Danes, French, we went to Dutch, you can't avoid Dutch. Look at twins and their spouses. We do see, once again, this uneven distribution, although it's less clearly bimodal, 19%, 1 out of 5 Dutch, but we selected them to be healthy. In these healthy guys, we see that these discriminant metagenomic species are very biased in distribution. The same algorithm that works for French and Danes works for Dutch as well, and bioclinical parameters go in the same direction. So we do see this huge difference in all of them. And when we try to interpret the microbiome, the potential of microbiome in low gene count individuals, then we see, say, this good friend of ours, Akkermansia, is low. Butyrate production is low, lactate is low, bifidobacterium seem to be low, and what is high? LPS producer, pro-inflammatory guys. So we do think that microbiome in low gene count individuals is less healthy. So then we did this kind of meta-analysis on about 1,600 people. Some people are healthy and have high gene count, most of them are above our threshold, which separates low gene count and high gene count. Dutch, Danes, we looked at Spaniards as well. I come back in a moment to these two. But when one goes to patients, say obese, type 2 diabetes, NASH, or IBD phenotypes, this is really lost. Type 1 diabetes is kind of exception, and it's an interesting exception. But in many of these diseases, this neglected organ seems to atrophy. So how about these two cohorts, we don't know whether there is a real biological signal or whether there may be some problem with it, because all of these guys were analyzed by the MetaHit pipeline that we developed. Well, there were some differences for the other two, and if you look at the quantitative metagenomics, then one can distinguish really two parts of it, DNA sequencing and bioinformatics, which we think was well standardized across these studies, but there were some differences, sample collection, processing, these two sides, and we just don't know whether it's a real loss or an apparent loss in these cohorts, and it's important to establish. Be it as it may, we think that standardization here is critical, and we really need to look into it. So, yes, we can detect the risk. But to prevent, we also need to alleviate. Can microbiome be a target for intervention? And we think about this microbiome perturbation not as a pathogen which comes in. It's not Ebola virus. It's kind of population change like eutrophic lakes. So can we restore microbiome? And we attempted to do that in microbees study, 49 overweight or obese French, which were following calorie-restricted diet for 6 weeks and maintenance diet for 6 weeks. That was done under responsibility of Karine Clément from Pitié-Salpêtrière. Jouy-en Josas was coordinator of this study. The dietary intervention, as I said, was calorie-restricted, high protein, low fat, and supplemented with fiber. What we observed, of course, is that all the metabolic parameters improved. That was expected. But what we also observed is that in low gene count individuals, this gene count was improved, although not fully. We also saw that high gene richness people do respond better to nutritional intervention. So that's a stratification between responders and non-responders. Another intervention in the context of IBS with probiotics, which we just got published. You know, probiotics, people eat these fermented meat products, so what's going on? We were able to find a number of metagenomic species, a small number of metagenomic species, which are impacted greatly by probiotic consumption. These metagenomic species, most of them had butyrate pathway modules. We look at the modules separately. Modules were clearly increased in the microbiome, and we also try to do to confirm that sort of observation by adding probiotics in the artificial gut system. We suppose there's lactate to butyrate conversion. So yes, we think we can impact in different ways microbiota. So one of the messages here is low diversity microbiome, low richness microbiome appears to be less healthy, can be diagnosed simply to identify individuals at risk, and interventions can correct it and possibly alleviate the risk, and this could have major impact on public health. Just as a reminder, chronic diseases, you know that, are frequent and costly. 4.4% of French population suffers from type 2 diabetes. Yearly cost is 12 billion euros. This is the deficit of social security in France, and it does reach 12 billion euros a year regularly. And the politicians try to do something about it, then it goes back then. So if we could retard chronic disease by systematic analysis and treatment of microbiota risk detection and alleviation, we could perhaps impact these things. But the question that often arises, and people ask me regularly, so that's why I'm going to address it directly, is change of microbial population cause or consequence of chronic disease? And I think it's a question for a lazy mind. And I think that it may very well be an irrelevant question, actually, because I think that people with lazy minds forget that there is yet another alternative, and it is that altered microbiome could contribute to the disease, and there is likely no single cause for chronic disease. And that you know as clinicians, so it's totally unfair to ask this stupid question. Okay. So let me give you the two examples which I have presented. How could microbiome contribute to the disease? Low richness gut microbiome may be less healthy. There are lower butyrate producers, and that could impact gut health. There are abundant pro-inflammatory species. So the systemic inflammation, which clinicians tell me, is a facet of almost all, if not all, chronic diseases. My other example is liver cirrhosis, where we think that this gut microbiome may be toxic by producing factors which impact encephalopathy. And you know, doctors treat these guys by giving them laxatives, okay? And they feel better, so you have even an experiment, an intervention. They feel better. Now we understand why they would feel better. So how could that less healthy, toxic microbiome, what's the advent of that? So I'm going to present two scenarios to you. One is what I call a vicious cycle scenario. And that, I think, could be the case in low richness people. A trigger could be years away, who knows, maybe a pathogen infection, food, pollutants, causing a modest inflammation or circumscribed inflammation in intestine. At that zone, we know that as microbiologists, inflammation will select for species which tolerate inflammation, which can detoxify raw species, which can repair their DNA. So that's your proteobacteria, which in turn produce lots of LPS and promote inflammation. So here you have a cycle. Inflammation, select for species which promote inflammation, and then you get this vicious cycle which we need to break. Result, pro-inflammatory microbiome which predisposes or aggravates chronic disease. My second scenario is loss of barrier scenario, and I think that this may well happen in liver cirrhosis, where a trigger could be virus infection, we know about that, alcohol, obesity, autoimmunity, parasites, other things. But that, we postulate, leads to impaired bile production, and that has been established. We don't discover it. But as a result, we propose as a model, barrier is lower, and then these oral bacteria and foodborne pathogens that we also find can now get into this niche and then synthesize all these molecules which are not good for us. So I think that we should strive to restore or preserve health by modulating unhealthy, toxic microbiome. And please don't misunderstand me. I think that we need to understand the mechanism by which we happen. But please change the paradigm. It's not cause or consequence, it's contribution, and let's try to understand how it works and help the patients by doing something about it. And I think that Willem also pleaded for that. But how to introduce microbiome into public health? Well, I'll tell you a few words about Metagenopolis, which was conceived by our government, who gave us 19 million euros for a period to try to address these questions, make this technology available for academics, medics, industry, try to unravel microbiome host links in human health, of course, but also in modern domestic animals. I work for INRA, so INRA, you know, agricultural institution worries about animals as well. And there are a number of applications. So what is Metagenopolis? Platforms and the clinical interface. Our clinical interface is PTSL Petrier Hospital via ICON program that Karine Clément heads, one of the biggest hospitals in Europe, so access to patients and clinical studies. Another of our founders is Catholic University of Lyon, who helps us for ethical and societal issues, because we do think that what we do can have great impact on the society, and we'd rather be advised and helped in that. The 3 platforms that operate, 4 platforms that are operated by INRA, one has to do with samples, biobanking procedures, automation, standardization. Our objective is to have a biobank of over 1 million stool samples, and that's just a very small pilot study if you think about 400 million or so of Europeans around here. We, of course, are integrated with biobanks in France. One of the big hurdles is big data analysis, and we have standardized that, industrialized that, so it works quite well. Why big data? Well, typically in a study such as the ones I have described, generate about 4.5 terabytes of data, and we can handle that in days, although biostatistical analysis take a little longer. We also have a functional metagenomics to go towards the mechanism. What is responsible for the dialogue? And since we don't know how to culture all these microorganisms and waiting, well, Willem can cultivate some of them, we clone their DNA. We clone like 40 kb, 40 genes. In E. coli, we have a library which contains about 13 million genes, and then we can screen the activities of the genes carried in these libraries by putting them, or rather lysates, in contact with eukaryote cell lines. We have about 16 screens, immunity, proliferation, metabolism, and others, and identified a number of bioactive clones that are good candidates to go further. Seeing that we are at the cutting edge in metagenomics with a number of visible papers, but that's very pleasant for a scientist, of course, and it's good to see that, and our friends are, jealous a little, so it's kind of fun. But it's not enough, because to impact the society, you have to engage industry. And we are doing that actively by founding Etheron, which has as a mission described here, and we do expect to have out this bacterial richness assessment kit by the end of the year, basically, which should help in going further. So I do think that human microbiome has great potential to impact public health, diagnostics, prognostics, patient monitoring, target for modulation to improve health. Of course, that's what we would like to do. And I think that it could help shift the focus of medicine, which is mainly curative today and too costly, to preventive tomorrow, and thus save not only untold resources, but also untold human suffering. So I just need to acknowledge many good friends, and you see some of them around here, like this guy you may have seen before, and this guy you'll see in a little while. Microbees, collaborators, all the Metagenopolis team, without whom nothing would happen. And I'd like to end by recommending you to take good care of your microbiota so it's rich, as this wonderful Japanese garden, rich and beautiful. And as a very final slide, I'd like to invite all of you to the next big event in human microbiome, International Human Microbiome Congress, which will be held next year in Luxembourg. Please come. Thank you very much.
So I actually believe every question is valid, and sometimes the really stupid ones are the really good ones. So in that sense, please ask questions. The first here.
You nicely sidestepped the cause and consequence question, but really contribution can go from 0 to 100%, at which one end is 0% is a consequence and 100% is a cause. So you're really looking at a continuum. So can I ask you then, rather than cause or effect, where do you think the contribution sits on the 0 to 100% scale?
Well, thank you for the question. I think it's one of the right ways of looking at it, because you are helping me escape this dichotomy, which I think is not only, well, I'd say it's really counterproductive, because if we are expected to show something which never exists, like a single cause, we'll all be blocked, and that's not what we should be doing. Like Willem said, you know, I'm getting old, I'm in a hurry. I want to see these things happen. So, you know, let's not be distracted by pursuing a very elusive goal. Let's do something concrete. And of course, try to find out how things work.
So, Frederick.
Thank you, Dusko. It was really great. I have a question about your high and low gene counts, because as you know, that it's a big difference in social economic status if you're healthy or if you're sick. So have you taken social economic status into consideration when you do these high and low gene counts?
No, we didn't. Thank you for the comment, Frederick. As I said, there may be many, many causes, and among the list of things I put down is diet, and it could be exposure to pollutants or to whatever you have as initial trigger. But it would be certainly worth looking into that.
I mean, this is usually related to
yes.
diet and social economic status
yes.
and microbiome.
Absolutely.
So here's the next question.
Okay, I now realize I have a lazy mind. So I'm going to start off my comment with a quotation from Cromwell. He was addressing the assembled Church of Scotland, and he addressed them as follows: "Gentlemen, in the bowels of Christ, I beseech thee, think it possible ye might be wrong." Now, what I'm saying is that where it is possible to study something, an effect that contributes to a disease, you're ethically obliged to do it. Now, I don't expect you to show an association between an event and a complex multifactorial disease. I never expect that. But what I do expect is that where you find an association between something and a phenotype, the association is of itself insufficient to be translated into public health medicine unless it is verified. That's my comment.
Well, thank you for the comment. You know, just to start with it, lazy mind is not a permanent syndrome. That's why I'm talking about it, to try to get people out of that frame of mind, because it's much too simplistic to say, show me the cause, because it may not be there. But I subscribe fully to what you say. At present, we are building these models. Now, in liver cirrhosis, we can see factors which clinicians with very diligent and careful work has established impact this phenotype, this comorbidity, this complication of liver cirrhosis. All we are saying at present is watch out. We have signals that these factors may be due to the microbiome. So what would you like us to do? Intervene? Okay. These interventions, very rough and say mechanistically not documented interventions, I give you laxative. That will chase out this microbiome, and you feel better. So, you know, if I wanted really to push my case, I said, you have a demonstration, give me a break. Now, what happens is that, of course, this toxic microbiome re-establishes itself. So I would plead with clinicians, let's try to do an intervention like Max does, okay? And try to substitute this unhealthy microbiome in people with a more healthy one. And then, to prevent its deterioration, maybe one should also consider medication which would restore the barrier. With bile salts or bile salts equivalent, there's a list of medications that could be tried. And I'm not saying that it should be done now with everybody, because, of course, you as a medical doctor know much more about it than I. I can just suggest maybe that would be a way to approach in a totally different way what is going on there. And beyond that, beyond that, we do need these longitudinal studies. We are trying to see whether indeed decompensation results from accumulation of these signatures. And we are trying to do these things. You see, we are opening things. I'm not advocating that everybody should go on that, but I would like to do myself whatever I can to try to screen people at risk with low richness, simple tests, cheap tests, maybe out of their pocket, or maybe whoever could help with that, and use that to try to improve their health, without waiting obligatory to show, yes, indeed, if you have more LPS-producing bacteria, you are likely to have more inflammation. 15 years later, I can show that it's bad for you. It has been shown.
I think we all agree there's still a lot of work to be done. I'm a trained immunologist, and I think to this day I could not tell anybody what's a healthy immune system. So I think we have to be careful with this. Next question.
In the example of hepatic encephalopathy, biochemically it is accumulation of aromatic amino acids, tryptophan, and serotonin that gives you the comatose state. Are you saying that in those microbiome changes happened before or after the change in the amino acid profile?
Okay. I did not really refer to aromatic amino acids because I don't know very much about them. I have referred to elements that the clinicians tell me are important for them. All I can say is that we do see a potential for production of these molecules in this microbiome. If these molecules are known to contribute to the disease, and if you can help me making my list longer so we can go and look at that, all I would say is, yes, there is a toxic element in your body, in body of a cirrhotic patient, which does generate these molecules.
So 2 last questions here, and then Adam has the final question.
Yes. Yeah. Hello. In the multifactorial conception of disease, clinical and epidemiologic data support that psychological stress might be involved in the onset of many diseases. So my question is, how would you think psychological stress could affect microbiota richness, and which could be the mechanisms between psychological stress and alterations of microbiota?
Well, my very quick answer is I don't know. And my little longer answer is, can we set up a study where we would look at people at stress and see, you know. I'm aware of some animal studies where, okay, a guy who put a mouse in a plastic bubble in a cage, let a big, huge rat run around it, so the mouse is very stressed. You know, one can induce that without much intervention and look at the microbiome. I'd willingly talk with you or whoever might be interested to try to find out what would be the best model to see impact of stress on evolution of microbiome. And, you know, Willem is much more intelligent than I am and knows much more and is young and has energy, so he can pull out a model. I could pull out a model also, but, you know, I'm lazy. Not of mind.
The final question, Adam.
I also have a question about public health. You've drawn a connection between the richness of the microbiome and the number of chronic diseases, BMI, obesity, type 2 diabetes, cardiovascular disease, even dental caries. And the recent question about stress is exactly pertinent. All those diseases, as well as stress, are tied to socioeconomic status, and they affect the poor much more than they do the rich. So my question is, what is the link between the richness of the gut microbiome and the actual richness? How is it predicted by socioeconomic status and incomes?
Yes, thank you. I mean, sometimes I say it, it's better to be rich than poor. Okay? Now, socially, one would tend to agree, and microbiotically, we seem to have the evidence for that. I can't really go beyond what I have already said. We have found some association with long-term dietary patterns, like people who eat more fruit and vegetables tend to have higher richness. We have not been in a position to estimate correlation with socio-economic status in there. But people kind of know that they're more obese, metabolic syndrome guys among. So it can all go together. It's, yeah.
Okay.
Thank you.
So I hate to cut short this discussion because there are lots of very interesting topics which came up, but we have to move on. So it's a pleasure to introduce Hiroshi Kiyono from the University of Tokyo. So he has a degree in dental sciences from Nihon University and then got his PhD from the University of Alabama at Birmingham. He one of the grandmasters of mucosal immunity and works at the Max Planck Institute, Osaka University, and now at the University of Tokyo. And he is currently the Dean of the Institute of Medical Sciences at the University of Tokyo and has made major contributions to mucosal immunity.
Thank you, Joseph. Before I start my presentation, I would like to take this opportunity to express my sincere appreciation to Thomas and their colleagues for organizing this meeting and inviting me to participate and share some of our recent progress for understanding of the gut immune system and commensal microbiome. Since we are behind the schedule, then we have 15 minutes to go for the lunch. So probably I have to speak faster than Dr. de Vos. So since I came from Japan, maybe I have to go to the speed of bullet train instead of express train. But anyway, as we've been hearing that our gut is enriched with commensal microbiota, then Dr. de Vos indicated that more than commensal right now, commensal meaning the sharing the food, sharing your table, but based on his presentation, probably beyond just sharing food and the table in terms of the interaction between the commensal microbiota and mucosal immune system. Now, as I illustrated in this slide, commensal microbiota provide beneficial molecule and nutrient for the host, including the immune system, shaping up immune system, maintaining appropriate immune system. But once this mutual system disrupted, some of the commensal microbiota can be triggering factor for the diseases and the susceptibility to the infection. And it is well known that commensal flora can influence on development of mucosal immune system or gut immune system more than 30 years ago. Original work was done by Dr. John Siebler. He nicely showed that commensal flora is triggering factor for the development of the mucosal immune system. So as illustrated in this slide, if you maintain mice in a germ-free condition in terms of the shape of the organized lymphoid structure, such as gut-associated lymphoid tissue or Peyer's patches, they are very immature in terms of size, in terms of the immunological function. But once you expose some of the commensal flora, E. coli, or E. coli-driven LPS into these germ-free mice, you start to notice that development of the organized lymphoid structure, such as Peyer's patches, leading to the increase of IgA B cells, and also at the same time, gut immune system develop oral tolerance. So intestinal commensal microbiota are essential for development, maturation, and maintenance of the appropriate gut immune system. So, as I indicated, if you kept mice under the conventional condition, you have high level of secretory IgA. But in case of germ-free mice, this secretory IgA is almost nothing. Today, I would like to discuss with, in terms of the interaction between immune system and the commensal flora, then, as you may know well, newly identified so-called innate lymphoid cells are currently central focus for the interaction between the commensal flora and host via epithelial cell. And these innate lymphoid cells now can be divided into three groups based on the necessity of transcription factor for the development of different subsets, and in terms of the cytokine production patterns, and also in terms of the function. And today, I would like to focus on group 3 or type 3 innate lymphoid cell in terms of regulating epithelial cell to create a commensal niche for the commensal bacteria. A few years ago, when we examining commensal flora and the interaction between host immune system, we decided to look at gut-associated organized lymphoid tissue. Paris patches are important site for the initiation and regulation of gut immune system. So we decided to look at the interaction of Paris patch and commensal flora. Then when we examined commensal flora on the dome epithelium of the Paris patches, we generally found so-called segmented filamentous bacteria. Now, as you may know well, these dome epithelium, well, Nestlé is saying quit, time for lunch. So anyway, this dome epithelium of the Paris patches containing unique antigen sampling cells, so M cell. So we thought that segmented filamentous bacteria may be uptaked by the M cell. Then when we examined inside of the Paris patches, in our surprise, we couldn't find any SFB. Instead, as illustrated green or yellow dot, alkaligenes were identified in the inside of the Paris patches. Then these alkaligenesis labeled with green fluorescence was given to the intestine. We were able to show that M cell taken up these alkaligenes inside of the Paris patches. Then three-dimensional analysis also showing that alkaligenesis can be uptaked by the M cell. Further, after uptake by the M cells, as illustrated in this slide, some of alkaligenesis can be taken up by the dendritic cell. Then we have now preliminary data suggesting that somehow these alkaligenesis can be escaped for antigen processing from dendritic cell and creating cohabitation niche in the inside of the dendritic cells. This point was further confirmed by the simple analysis of alkaligenesis specific antibody responses. So when we examine mice kept under the SPF condition, we could detect alkaligenesis-specific secretory IgA, but not germ-free mice. Further, when we analyzed serum sample from SPF and germ-free mice, although secretory IgA antibody against alkaligenesis can be found in the fecal sample, we couldn't find any alkaligenesis-specific serum IgG antibodies. So this suggests that somehow our gut immune system contains alkaligenesis inside of the pilus patches. The presence of alkaligenesis in the inside of the pilus patches has been also confirmed in the primate and the human. So if we examined pilus patches from healthy monkeys or pilus patches from non-inflamed tissue of the human, we could identify alkaligenesis, and also most of the us containing alkaligenesis-specific secretory IgA antibodies in the fecal samples. So as monkey as well. So these results suggest that some of the commensal flora can create cohabitation niche in the inside of the organized lymphoid structure. So in the next, we examine how these intertissue cohabitation can be contained in an organized lymphoid structure. Together with David Alice's group at Penn, we are able to show that so-called innate lymphoid cells seems to be an important player for the containment of the alkaligenesis in the inside of the pilus patches. So if you delete alkaligenesis, if you delete innate lymphoid cell, we notice that innate lymphoid cell expressing CD90, so if you inject anti-CD90, you can abrogate or you can remove innate lymphoid cell. And when you create these conditions, we start to notice that alkaligenesis can be found in a systemic compartment, such as spleen and liver, which resulted in induction of severe inflammation in a systemic compartment, such as spleen. These findings were also confirmed in the human level when David and his colleagues compared patients with pediatric Crohn's disease and control group. They found that higher titer of alkaligenesis-specific IgG antibodies patient with inflammatory bowel disease. So these results suggest that alkaligenesis may be involved in the creation of homeostatic condition, and removal of alkaligenesis may leading to the initiation of intestinal inflammation. These findings were further confirmed by our study together with Dr. Hideki Iijima's group at Osaka University. We examined adult population. As I indicated, in a healthy condition, you can find alkaligenesis in the inside of the pilus patches, but some of the Crohn's disease patients, we noticed that significant reduction of alkaligenesis, or they are gone from the pilus patches. On the other hand, in terms of the antigen-specific IgG antibodies, as like pediatric case, higher level of IgG antibodies against alkaligenesis was noted. At the same time, level of secretory IgA antibodies against alkaligenesis was decreased in a patient with Crohn's disease. So alkaligenesis may be important for the creating the symbiotic condition for the gut immune system and outside environment. We recently succeeded to create alkaligenesis mono-associated or notobiotic mice. When these notobiotic mice were subjected to the DSS-induced intestinal inflammation compared with E. coli mono-associated mice. The E. coli mono-associated mice resulted in severe inflammation in intestine, then some of mice died. But in case of alkaligenesis mono-associated mice were protected from the DSS-induced inflammation, suggesting that alkaligenesis could be an important cell for maintaining appropriate coexistence of the commensal flora and mucosal immune system. So, this is a result clearly indicating that some of the commensal flora can be creating the cohabitation niche in the inside of the pilus patches, and innate lymphoid cells provide cytokine, so-called IL-22, which are potent cytokine for regulating epithelial function. And I did not show you data, but we have evidence that these signaling cascade induce antimicrobial peptide, which may be involved in the containment of the alkaligenesis in the inside of the pilus patches. So, first part of the study, we show that innate lymphoid cell producing IL-22 may be important for the creation nerve, intratissue habitation nerve, some of the commensal flora, alkaligenesis. This study has been carried by the Dr. Takashi Obata, who is now at the Takeda Pharmaceutical Corporation. When he was a graduate student with us, he mainly carried out this project. In the second part of the talk, I would like to share some of our newest data showing that these ILC play important role glycosylation of epithelial cell, which also provide cohabitation niche for some of the commensal flora. This study has been carried by the Dr. Yoshiyuki Goto. His nickname is Gochi, who is now in the Columbia University in New York as postdoctoral fellow. Then fucosylation of the epithelial cell has been shown to be important for the creation of cohabitation niche. For example, Dr. Hooper's group nicely showed that some of the commensal flora produce fucosidase to cleave alpha-1 to fucose, and these alpha-1 to fucose are important nutrient for some of the commensal flora. In order to induce fucosylation, FUT2 is an important enzyme. Then FUT2 mutation has been associated with several diseases as well as susceptibility to the infection, as illustrated in this slide. For example, FUT2 nonsense mutated people has been shown to be resistant to norovirus and rotavirus infection. Further, FUT2 nonsense mutation has been shown to be associated with the incidence of the Crohn's diseases. So, fucosylation and inducing enzyme FUT2 seems to be very important for the creation of symbiotic condition as well as pathologic condition. So we are wondering how these FUT2 expressions by epithelial cells are induced and regulated. So Gochi decided to look at fucosylation or fucosylated epithelial cell using Murin system. So he simply divided small intestine into 4 parts from duodenum to ileum. Then he examined level of or number of fucosylated epithelial cells. As illustrated in the slide, in case of Murin system, most of the fucosylated epithelial cells were associated with part 4 or ileum. So when he isolated cell, high number of fucosylated epithelial cells can be found when compared with epithelial cells isolated from the duodenum or part 1. In terms of the level of FUT2 expression, correlate with number of fucosylated epithelial cell. So when he isolated epithelial cell from the part 4 or ileum, high level of FUT2 expressions can be observed when compared with part 1 or duodenum. So this is suggesting that commensal flora may be involved in the induction of FUT2 expression. Then, as we heard, based on the Joseph's nice introduction, gut consisted with large number of different types of immunocompetent cells. So Gochi next decided perhaps some of the lymphocytes located in the lamina propria region of intestine may regulate FUT2 induction. So he tested different gene manipulated mice. Among them, ROR gamma knockout mice, which lacking Th17 cell and innate lymphoid cell. Also, these mice has been shown to defect in the secondary lymphoid organ development, including Peyer's patches, and also reduction of IgA. So, which subset of cells are involved in the regulation of fucosylation? Now, as I indicated, wild-type mice and ROR gamma knockout, which harbor these deficiencies, no fucosylated epithelial cells can be found in a ileum, or part 4. So in terms of the number of fucosylated epithelial cells are significantly reduced, and level of FUT2 expression was also significantly reduced in these mice. So these results suggesting Th17 cells, ILC, organized lymphoid structure, or IgA-producing B cell may be involved in the induction of FUT2 expression. So Gochi decided to look at first T-cell deficient mice, including Th17 cells. These T-cell deficient mice possess normal level of fucosylated epithelial cells. So probably Th17s are not involved. Next, he examined B-cell deficient mice. again, showing normal level of fucosylation or fucosylated epithelial cell. So probably B cells are not involved. In terms of organized lymphoid structure, we can create Peyer's patch deficient mice by injecting anti-IL-7 receptor antibody. Again, deficiency of Peyer's patch still maintained normal level of fucosylated epithelial cells. So we thought innate lymphoid cells could be key cells for the induction of fucosylated epithelial cells. As I briefly indicated, these innate lymphoid cells possess unique transcription factor requirement, ID2 and ROR gamma, for their development. In terms of the phenotype expression, they preferentially produce IL-22, and they express CD92, CD127, and so on. So next, Gochi decided to examine ID2 knockout mice, which lacking IL-C. As illustrated in this slide, ID2 deficient mice, we couldn't find fucosylated epithelial cells. Next, using an in vivo system to inject anti-CD90 antibody to deplete these IL-C. As illustrated in this slide, IL-C express high level of CD90. So we treated mice with anti-CD90. Then control group treated with isotype control antibody, which resulted in removal of innate lymphoid cells, resulted in reduction or lack of fucosylated epithelial cells. So these results clearly indicating that IL-C is key population for the induction regulation of fucosylated epithelial cells. To further prove this, we tested LAG1 deficient mice. Based on the study done by earlier Dr. Sawa's group, or Dr. Sawa and his colleagues at Pasteur, showing that LAG deficiency resulted in increased level of innate lymphoid cell. So when we examined in terms of fucosylated epithelial cell, in a part 4 or ileum, you notice that increase of fucosylated epithelial cells. Even part 1, duodenum, generally they are negative for the fucosylated epithelial cell, but increased number of IL-C resulted in induction of fucosylation in part 4 or duodenum, further suggesting that IL-Cs are important cell population for the induction regulation of fucosylation of epithelial cells. In terms of the key cytokine, as I indicated, IL-C, especially type 3, preferentially produce IL-22. So we confirmed that when we isolated different subsets of cell, IL-C3 produce preferentially IL-22. Then LAG1 knockout mice, which possess high number of IL-C, were treated with anti-CD90 antibody, which deplete IL-C3, resulted in no production of IL-22. Further, IL-22 production by ILC3 seems to be regulated by the commensal flora. So if you treat mice with antibiotics, we start to notice that reduction of IL-22 production by ILC3. To further prove IL-22 is key cytokine for inducing FUT2 expression for the phosphorylation of the epithelial cells, Gochi used hydrodynamic tail vein injection of plasmid containing IL-22 gene. So when these two groups of mice were treated with empty plasmid and plasmid containing IL-22, then we noticed that level of course, IL-22 went very high. Then when we examined these mice, we noticed that induction of ghost-related epithelial cell, even in a part 1 duodenum, in a physiological condition, you won't find these ghost-relation, suggesting that overdose of the IL-22 can induce ghost-related epithelial cell, showing that critical importance of IL-22 mediated signaling cascade. So when we examine IL-22 knockout mice, level of ghost-relation reduced, and FUT2 expression level by epithelial cells was also reduced. So far, our data clearly showing that innate lymphoid cell producing IL-22, which act on epithelial cell to induce FUT2 expression, leading to the induction of ghost-related epithelial cell. So if ghost-relation of epithelial cells are gone in the intestine, what will happen? So Gochi examined in terms of the FUT2 deficient mice compared with control mice, we noticed that profile of the commensal flora changed. In terms of SFB, segmented filamentous bacteria, which attach to the epithelial cell, didn't change, but Lactobacillus was reduced in mouse lacking FUT2 or ghost-related epithelial cells. In terms of the susceptibility to the infection, FUT2 deficient and control mice are infected with Salmonella typhi murine. Both mice are infected with Salmonella typhi murine, but the severity of disease was much higher in the FUT2 deficient mice, and number of Salmonella typhi murine in the cecal tissue is much higher in the FUT2 deficient mice, suggesting that lack of FUT2 or FUT-ghost-related epithelial cell increase susceptibility to the Salmonella infection. So our findings suggested that ILC type 3 or group C ILC are key cells for the regulation of FUT2 expression by epithelial cell via IL-22. These ghost-related epithelial cells contribute for the creation of normal commensal niche, and also important for some of the infection, in this case, Salmonella, for protective barrier. Since our study was published in the summer, recently, two similar papers came out in Nature and Cell Host Microbe. by Dr. Cherabonsky's group at the University of Chicago, and also Dr. Lowry's group at Sanger Institute, UK. In addition to our findings, they have shown that LPS can stimulate dendritic cell to produce IL-23, which stimulate ILC3 to produce IL-22. Further, disruption of FUT2 expression leading to the just not S-type murine, C. rodentum infection susceptibility were also increased and also created this biosis lacking of the fucosylated epithelial cells. So these results clearly indicating that, as you might expect, glycosylation of epithelial cells are very important for the creation of commensal niche and also protection against pathogens. So finally, I would like to acknowledge our colleagues who have been involved in this project. Especially, I should acknowledge Gochi, Yoshiyuki Goto, and Takashi Obara for the alkalogenesis study, as well as Group 3 ILC regulated glycosylation of epithelial cell, together with excellent collaborator in Japan, as well as the rest of the world. Thank you very much for your kind attention.
So, questions?
In Crohn's disease, the question of modification of intestinal bacteria is still largely debated. In your model, you consider that alkaligenase is inducing inflammation. My question is, what is the real evidence that the presence of alkaligenase is not secondary to the inflammatory process?
I think that's an excellent question. We still don't know. As you pointed out, it could be because of the result of inflammation, alkalogenesis might be gone, or lack of alkalogenesis may disrupt symbiotic condition leading to the inflammation. So we are still looking at both possibilities. But in terms of the not-biologic mice study, although it is a still preliminary result, it seems that monocolonization of alkalogenesis in mice resulted in less susceptibility to the DSS-induced colitis induction. So our current working hypothesis is that alkalogenesis may inducing more regulatory type environment, including regulatory T cells in the inside Paris patches, which might be contributing downregulation of inflammation.
Thank you for your beautiful talk. I also like the bullet train thing. I will talk slowly. I have a question about the alkaligenase, and the reason is that the question is that can you isolate it from the Paris patches in a live form, and how does it look like? Because some alkaligenases actually are capable of reducing nitrite and making NO and other compounds which are actively signaling. So I'd like to know what you have done there.
So we've been trying to isolate alkalogenesis from Paris patches. We have been not successful. So we are using that in a monocolonization study using alkalogenesis fecalis, which are already culturable microorganisms. So that point, answer is we still cannot isolate alkalogenesis from the
But
Paris
do you know
patches.
they're alive, or can it be pretty dead there?
We have,
do some staining to see whether they're alive or dead.
yeah, I think, our preliminary results show that they are alive.
Okay. So, next question.
Thank you for a very insightful talk. My question is about the role of dysbiosis induced by the mutation of FIL2 in increased susceptibility to salmonella infection. You show the reduction, for example, reduction of lactobacillus. But if you replenish lactobacillus, can you stop the infection? And do you have any evidence it's causal or not?
That's also a good question, too. So we still don't know why FIL2 deficient mice lacking lactobacillus or reduction of lactobacillus. As I indicated in my introductory slide, Dr. Hooper's group showed that some of the Bacteroides can produce fucosidase to cleave glycosylated fucose to mucus layer. And these alpha-1, 2 fucose has been shown to be key nutrient for the Bacteroides. And whether lactobacillus use alpha-1 fucose as a nutrient for their growth, we still don't know. Then it is interesting that, as you suggested, if we reintroduce lactobacillus into FIL2 knockout mice, whether we can enhance fucosylation or enhance protection against salmonella need to be examined.
So I was just told lunch is waiting, so forgive me, I will only accept the final question, so please. Then we'll go for lunch.
The innate...
Forgive me, everybody else who want to ask, sorry.
The innate lymphocytes that you speak of, it's a really number of different kinds of cells, NKTs, gamma delta Ts, and they secrete a lot of cytokines. What's going on? I mean, you mentioned IL-22 and IL-23, but what about the rest of the environment there and the pattern of cytokines that might be present and affecting this? Do you know?
So, in terms of the other, two subset of innate lymphoid cell, whether they are involved in IL-22 mediated fucosylation, the other two subset doesn't produce IL-22. Then only group 3 ILC preferentially produce IL-22. So we think that the other two subsets are important, but not involved in induction of FIL2 by IL-22 signaling.
Thank you.
Thank you. Thank you.
So it's lunchtime now.